Good afternoon, students.
Good afternoon, ma'am.
Good afternoon, ma'am.
Good afternoon, ma'am.
Good afternoon. আচ্ছা, তোমাদের তো এখন সেমিনার কিছুক্ষণের মধ্যেই স্টার্ট হবে, তো বি কনফিডেন্ট। তোমরা যেমন পারবে তেমনই বলে যাবে, ফাম্বল করবে না, ভয় পাবে না, আটকে যাবে না, ঠিক আছে? সুন্দর করে বলে যাবে। সবসময় মনে করবে তুমি যেটা বল
হ্যাঁ,
সবাই আসলে এখনো আসেনি।
হ্যাঁ, আমি ভিডিওটা অফ করে দিচ্ছি। হ্যাঁ,
আপনার আমি বলবো।
হ্যাঁ। তোমাদের ডিপার্টমেন্ট সবাই এসছে?
ডিপার্টমেন্টে অর্পিতা দিয়ে এসছে, অর্পিতা এসছে, দেবশ্রী দিয়ে এসছে।
আচ্ছা,
তো ওনার শরীরটা একটু খারাপ হয়েছে।
কার?
টিএস এর প্রফেসর।
আচ্ছা,
শরীরটা একটু খারাপ হয়েছে, তো হয়তো জয়েন করতে পারবেন বলে মনে হচ্ছে না।
আচ্ছা,
আপনি সপ্তাহে জয়েন করবেন।
আচ্ছা, আচ্ছা। এতজন তো ভিডিও অন করলে হবে না, তাই না? আমি
হ্যাঁ হ্যাঁ, না আমরা যখন মানে ডিউরিং লেকচার সেই সময়টা আমরা অন করবো, আবার ছেলেমেয়েরা তো সবাই নিজের প্রেজেন্টেশনের সময় on করবে।
আচ্ছা, ঠিক আছে। এই কথা শোনা যাচ্ছে রে?
হ্যাঁ দিদি, পরিষ্কার।
আচ্ছা, সেটাই। প্রিন্সিপাল ম্যাডাম জয়েন করবেন তো?
হ্যাঁ ম্যাডাম জয়েন করবেন, করেন আমি টক করবো একবার।
আচ্ছা।
পুষ্পিতা দি, গুড ইভিনিং, ওলি বলছি।
হ্যাঁ, দেখতে পাচ্ছি না বলে আর কি। হ্যাঁ, ঠিক।
ভিডিওটাকে অফ করে রেখেছি আর কি, এতজন।
ভালো করেছিস। হ্যাঁ, হ্যাঁ, হ্যাঁ।
ভিডিওটাকে ভালো আছো?
হ্যাঁ, হ্যাঁ, ভালো আছে। এই তোরা তোদের ডিপার্টমেন্ট আমাকে যা মানে যা খাতির করে, মানে যা রেসপেক্ট
না,
করে,
এরম বলো না।
এইটা
তুমি আমাদের উৎসাহ যোগাও, আমাদের
না,
ভালো লাগে সেটা।
মানে আমি ভীষণ সব সময় এটা নয়, মানে যে কোনো তোদের সেমিনারে আমি দেখেছি, আমি খুব আমি অত্যন্ত লজ্জিত যে এইভাবে আমাকে
এরকম বলবে না, আমরা অত্যন্ত গর্বিত তোমাকে পেয়ে, সব সময় পেয়ে, সেটাই চাই আমরা আগামী দিনেও।
তোরা খুব আমাকে ইয়ে করছে, আমি দেখেছি, আমার খুব নিজেকে ইয়ে লাগে।
থাকলে তো আমাদেরও ভালো লাগে না। আর এই তো অনলাইনে তো সবাই ছাড়াছাড়ি না, এই একটু সুযোগ হয় দেখা সাক্ষাৎ। একটু কথা বলার, এই আর কি।
সেইটাই তো কথা। তো তোদের এত সুন্দর আয়োজন, এত সুন্দর আমি প্রতিবার খুবই ভালো লাগে।
অর্ঘ্য, অর্পিতা ওরা সব করে আয়োজন,
খুব ভালো পুরো ডিপার্টমেন্ট।
পুষ্পিতা দি,
হ্যাঁ বল বল।
তুমি সবসময় আমাদের সাথে থাকো এবং একটা সেমিনারে শুধু শুধু ছিলেই না, তুমি শেষ অবধি সবকটা শুনেও ছিলে। অলদো বটানি ছিল, বাট স্টিল তুমি কিন্তু ছিলে এবং সবকটা শুনেও ছিলে। আমাদের ভীষণ ভালো লাগে এই ব্যাপারটা।
আমি থাকি, আমি ভালো লাগে আমার, মানে আমার সর্বস্ব দিয়ে আমি থাকতে চেষ্টা করি এবারে সব কাজে। ভালো লাগে আমার আর কি,
সেটা আমাদের পরম প্রাপ্তি।
আর নতুন কিছু শিখছি তো খুব, মানে বেশ একটা, মানে বেশ অন্যরকম কত কি জানতে পারছি, এটাই ভালো লাগে।
এটা ঠিকই বলেছো, এই অফলাইন অনলাইনে হয়ে আমরা সত্যি সবাই অনেক কিছু জানতে পারছি।
আর আরেকটা কি বলতো, মানে যতই তোদের থেকে জানছি ততই মনে হয় কিছুই জানি না, তাই না? মানে এরকম আমার মনে হয় যে সত্যি কত কি জানার আছে, কত
আসলে জানার তো কোনো শেষ নেই, সেটা ঠিকই বলেছো, ঠিকই বলেছো।
আর এই যে তোদের ছাত্ররা সব বলবে রিয়েলি, মানে এই ধরনের সেমিনার কিন্তু খুব দরকার ছিল, খুব ভালো।
হ্যাঁ দরকার, এটা মাঝে মাঝে মানে করাটা, আয়োজন করা পরীক্ষার সামনে সত্যি খুব দুষ্কর, কিন্তু করতে পারলে খুব ভালো হয়, করতে পারলে হ্যাঁ ছাত্রদের দিক থেকেও খুব ভালো হয়।
হ্যাঁ ছাত্রদের দিক থেকে খুব ভালো। আর ওরা তো একটু অনলাইনে সত্যি কথা, মানে ওদের একটু এই ধরনের উৎসাহ দেওয়া, তোদের ডিপার্টমেন্ট খুব ভালো কাজ করছে, খুবই ভালো। মানে
আমাদের স্টুডেন্টরা সত্যি খুব এনথুসিয়াস্টিক, মানে ওদেরকে কোনো কাজ দিলে ওরা 100% সেটা করে। ওদের দেখে আমরাও ইন্সপায়ার্ড হই এবং ভাবি ওদের জন্য আর কি কি করা যেতে পারে।
ঠিক ঠিক ঠিক,
সত্যি আমাদের স্টুডেন্টসরা ভীষণ ভালো,
খুব 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 খুব, আমি তাই দেখছি খুবই ভালো। মানে আমার তো স্টুডেন্টরা তো আমাকে লিংক পাঠিয়ে দেয়, মানে সবেতে হেল্প করে। একটু যদি বলি ব্যাস ম্যাডাম এইটা করুন, এভাবে করুন, ওইভাবে করুন, মানে সব সময় ওরা সাহায্য করছে। মানে
আর এখন স্টুডেন্টরা এইসব ব্যাপারে খুব ওয়াকিবহল, আমার থেকে অন্তত বেশি, আমি যেটা মনে করি। আমার তো প্রত্যেকটা জিনিস বাড়িতে ছেলেমেয়ের কাছে শিখতে শিখতে বকুনি খেতে খেতে প্রাণ যায়।
আর সত্যি আমার তো কেউ মানে হ্যাঁ একজন আছে,
এখন তো রিজু তো বাইরে, খুব স্বাভাবিক।
এবার সবই ওই থেকে থেকে নিজে নিজে করে করে শেখা। সত্যি ভালো লাগে, খুবই ভালো লাগে, অন্যরকম হচ্ছে, খুবই ভালো লাগে। তোদের সত্যি তোদের এই ইনিশিয়েটিভটা আমার খুব পছন্দ, খুব ভালো, খুব ভালো।
খুশিয়াদি ম্যাডাম জয়েন করেছেন।
ওকে, হ্যাঁ
ওকে।
গুড ইভিনিং ম্যাডাম। গুড ইভিনিং ম্যাডাম, প্রিন্সিপাল ম্যাডাম, গুড ইভিনিং।
গুড আফটারনুন ম্যাডাম।
আছে
গুড ইভিনিং ম্যাডাম।
ম্যাডাম শুনতে পাচ্ছেন?
মনে হয় ম্যাডাম শুনতে পাচ্ছেন।
মিউট কর।
হ্যাঁ
ও ওই জন্য শুনতে পাচ্ছি।
ম্যাডাম আনমিউট করেছেন।
ম্যাডাম
গুড ইভিনিং ম্যাডাম।
ম্যাডাম শোনা যাচ্ছে?
শুনতে পাচ্ছেন না
মনে হয় না শুনতে পাচ্ছেন।
ম্যাডাম শুনতে পাচ্ছেন ম্যাডাম?
মনে হয় শুনতে পাচ্ছেন না সম্ভব।
নাহলে উনি রিপ্লাই দিতেন, শুনতে পাচ্ছেন।
আমি ওই জন্য ভিডিওটা অন করলাম যাতে দেখতে পান।
এবার একটু অন করি তাহলে।
হ্যাঁ
শুনতে পাচ্ছেন ম্যাডাম? আমি ম্যাডামকে একবার কল করি।
হ্যাঁ একটু কল কর তো আরো,
তাহলে বুঝতে পারবে।
উনি ঠিক মানে কিন্তু মিউট করা নেই তো দেখছি।
হ্যালো
হ্যাঁ এই তো এই
হ্যাঁ,
শোনা যাচ্ছে।
গুড ইভিনিং ম্যাডাম।
ম্যাডাম শুনতে পাচ্ছেন?
হ্যালো।
হ্যাঁ হ্যালো ম্যাডাম, আপনাকে শুনতে পাচ্ছি, আপনি শুনতে পাচ্ছেন?
গুড ইভিনিং ম্যাডাম, শুনতে পাচ্ছেন?
না, মনে হয় না ম্যাডাম শুনতে
পাচ্ছেন না,
শুনতে পাচ্ছেন না।
অর্ঘ, একটু দেখো তো কি প্রবলেম।
পুষ্পিতাদি গুড ইভিনিং।
হ্যাঁ, গুড ইভিনিং সুশ্রী।
হ্যাঁ, তোমাদের কথাবার্তা শুনছিলাম এতক্ষণ।
ও শুনছিলি? আচ্ছা।
তুমি চুপচাপ ঘাপটি মেরে বসে আছো কেন? শুনলে হবে, বলতে হবে।
আরে সবাই তো বলছে, আমি একটু শুনি।
আচ্ছা আচ্ছা,
সেই একই কথা আমারও। সবাইকে গুড ইভিনিং। আপনারা সিনিয়ররা এত কথা বলছেন তো সেখানে আমরা একটু শুনতেই পছন্দ করি।
বেশি বলে ফেলেছি।
না 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 ম্যাডাম, এই
Sorry.
বাবা এইসব বলবেন না প্লিজ। দেখুন আপনারা আমাদেরও মেন্টা, কেন এটা এরকম বলছেন?
না না, আমি ইয়ার্কি মারছি, এই তো একটু প্ল্যাটফর্ম ইয়ার্কি করার।
শরীর ঠিক আছে?
হ্যাঁ
হ্যাঁ দেবশ্রীদি,
শরীর ঠিক আছে তোমার?
বুঝতে পারছি না, চিন্তায় আছি, এখনো তো
এই তো
জ্বর কমেনি।
মাস্ক পরে বসে আছে।
এখনো জল আসেনি? এখনো জল আসেনি?
জল এলো এই সবে, কিন্তু আমার জ্বর এখনো কমেনি, ওইটাই চিন্তা লাগছে, দেখি কি করা যায়।
তুমি আজকের দিনটা দেখো, তারপরে কালকে ডিসিশন নেওয়ার নিও।
এই তো রাখা দি হয়েছে,
রাখা ম্যাডামও সাজুগুজু করে ভালোই চলে এসেছে।
গুড ইভিনিং রাকা।
এভরিবডি।
হ্যাঁ,
গুড ইভিনিং টু ইউ অলসো।
Where is the tea? Where is the tea?
সমুদ্রের কফি কোথায়?
রাখা কেন? আপনি কিন্তু ফুড মিনিস্টার, ভুলে গেছেন?
এই তোদের সবটা জয়েন করেছি, তোরা কিন্তু আমাকে চা খাওয়াবি পরে।
হ্যাঁ অবশ্যই, অনেকগুলো ডিউ হয়ে গেল, অনেকগুলো।
এত চা খাওয়াবো, বলবে আর চা খাবো না।
ক্লাস ভর্তি করে চা নিয়ে যাব।
শুধু কি চা,
কলেজে
চায়ের সাথে টাও তো আছে না?
নিশ্চয়ই নিশ্চয়ই।
ম্যাডাম কি হলো?
হ্যাঁ ম্যাডাম, মনে হয় অসুবিধা হচ্ছে ম্যাডামের।
হ্যাঁ
প্রবলেম হচ্ছে।
হ্যাঁ দেখা যাচ্ছে,
আমার
কথা শোনা যাচ্ছে,
শোনা যাচ্ছে, আপনি শুনতে পাচ্ছেন?
গুড ইভিনিং
আমার কথা শোনা যাচ্ছে তো?
হ্যাঁ হ্যাঁ শোনা যাচ্ছে।
আচ্ছা,
আপনাকে গুড ইভিনিং ম্যাডাম,
আপনি
আপনাকে গুড
শুনতে
ইভিনিং।
পাচ্ছেন ম্যাডাম?
হ্যাঁ গুড ইভিনিং।
হ্যাঁ গুড ইভিনিং ম্যাডাম।
দেখতে পাচ্ছি না আপনাকে।
আমরা দেখতে পাচ্ছি না ম্যাডাম।
ম্যাডাম ভিডিও,
আমার ভিডিওটা অফ করা।
হ্যাঁ
ম্যাডাম।
হ্যাঁ
এবার ঠিক আছে, এবার দেখা যাচ্ছে। আচ্ছা
হ্যাঁ ম্যাডাম, ক্লিয়ার।
ভালো আছেন ম্যাডাম?
হ্যাঁ
আচ্ছা ঠিক আছে।
ম্যাডাম আমাদের টিচাররা সবাই উপস্থিত, টিসিএস হিসেবে পুষ্পিতা দিয়ে হয়ে গেছেন, শুধু আমাদের তরুণ স্যার, ডক্টর তরুণ কুমার সরকার একটু অসুস্থ, সেই কারণে মোস্ট প্রবাবলি জয়েন করতে পারবেন না, হঠাৎই শরীরটা একটু খারাপ হয়েছে ওনার। তা সেই কারণে যেহেতু আমাদের ছাত্রছাত্রীরাও প্রায় সবাই জয়েন করে গেছে, তাই আমরা উইদাউট এনি ডিলে এটাকে আবার স্টার্ট করবো।
অর্ঘ এখন অফ করি?
হ্যাঁ দিদি,
এখন আমরা ভিডিও অফ করছি। হ্যাঁ
ভিডিওটা তুই যখন বলবি তখন আমরা।
ম্যাডাম তাহলে আপনার পারমিশন নিয়ে আমরা শুরু করছি।
হ্যাঁ শুরু করো।
ঠিক আছে ম্যাডাম। সমস্ত ছাত্রছাত্রীদের ওকে জানাচ্ছি যে আজ আমরা স্টুডেন্ট সেমিনার 2021, যেটা বটানি ডিপার্টমেন্ট বঙ্গবাসী কলেজ অর্গানাইজ করেছে, এটার অফিসিয়াল ওপেনিং সেশন হবে এখন। সেই ওপেনিং এর জন্য আমি রিকোয়েস্ট করব আমাদের এইচওডি ডক্টর অর্পিতা স্বর্ণকার ম্যাডামকে টু কাইন্ডলি ওপেন দ্য সেশন, প্লিজ ম্যাডাম।
থ্যাংক ইউ অর্ঘ। respected Principal Madam, Dr. Himadri Chakraborty Bhattacharya Madam, Teacher Council Secretary, Professor Pushpita Sarkar Madam, my esteemed colleagues, and my beloved students. Today we are here for participating in the Student Seminar 2021. Students of SEM 2, 4 and 6 Botany Honours students will be presenting their topics through PowerPoint presentation. I think this presentation in a seminar will enrich them and other participants and also boost their confidence. I hope all the students will be able to deliver their presentations in their best possible ways and utilize this experience in near future. PowerPoint presentation is a part of honors curriculum and included in DSc paper called research methodology. Long way back in probably 2008, I had also organized a student seminar in our department with the cooperation of all departmental teachers. I am happy that again we are able to organize student seminar in 2021. In future, we will think about student seminar with participants from other colleges also. Although it is a competition, but to me, all participants are winners. My best wishes to all of you. With these words, I open this session of seminar. Now I would request Dr. Oli Khan Shein to present the welcome address. Madam, please.
Thank you, Orpita. It is immense pleasure for me to welcome our dear principal, Dr. Himadri Chakraborty, head of our institution, Bangabasi College, to our department, Botany department. Her august presence will definitely make this seminar a great success. When our convener, Dr. Argha Sarkar, presented the proposal for holding the seminar, Principal Madam immediately gave the permission without any hesitation. I think this seminar will be an immense benefit to the students for getting valuable informations which will be very helpful in their future life. Thank you all. Thank you, Madam. I would also welcome our Teachers Council Secretary, Dr. Pushpita Sarkar. Her august presence is very much helpful to us. She always is present in our different programs and seminars and encourages us from time to time. Thank you. Thank you, Pushpita Di. Thank you all. Thank you all.
Thank you, Validi. Now I would request our Madam, Head of the Institution Principal, Dr. Himadri Chakraborty Bhattacharya, for her welcome address towards the session and towards the students. So, Madam, please. Madam, can you hear me, Madam?
হ্যাঁ
হ্যাঁ ম্যাডাম প্লিজ আপনি কিছু বলুন।
আচ্ছা হ্যাঁ শোনা যাচ্ছে?
হ্যাঁ ম্যাডাম পরিষ্কার
শোনা যাচ্ছে? আচ্ছা
হ্যাঁ
হ্যাঁ Good afternoon to all of you, head of the department, Madam Orpita Sarnukar, Oli Khan Madam, and all my colleagues of the Botany department, and our TC Secretary, Dr. Pushpita Sarkar. Good evening to all of you, and it is pleasure for me to be present in this seminar, which is the student seminar organized by the Botany department of our college, Bangabasi College. I think this is very rare seminar for I don't know. I have come recently, so I don't know if the other departments organized it like student seminar. So many seminars were organized before for the teachers and for the students by the invited speakers and the other speakers, but I don't know if were any seminars of the students where students are the speakers. So it is my pleasure to say that the students are the speakers and they will represent their knowledge, and the teachers can estimate how are they, how their knowledge has been developed, all these things, and it intricates the bonding between the teachers and the students. So it is definitely a very very pleasuresome, and I very much encourage this type of seminar. I think their knowledge will be enriched, the students knowledge will be enriched. And nowadays in the CBCS system, our syllabus, there are so many DAC and ACC parts in our syllabus which were introduced from 2018. We are following that syllabus, and based on that syllabus it is important for the students to deliver some seminars and to write something and to speak something about their innovative. It is it is a culture of the innovativeness within the students. And so uh, I'm thankful to the department, and I also encourage it. Now you can start this seminar, and uh, hear my good wish to all of you.
Thank you so much, madam. Thank you for your enlightening lecture. And exactly, madam has sensed the essence of the student seminar. She has beautifully explained what is the utility of the student seminar. So I would forward to the next event. And now, as a convener, I would request our esteemed colleague, Mrs. Rakha Dasgupta, Rakha madam, to say about the utility of student seminar. আর এই স্টুডেন্ট সেমিনারের কি প্রয়োজনীয়তা, কি কারণে আমাদের এই অর্গানাইজেশন, কি কারণে আজ সমস্ত স্টুডেন্টরাই প্ল্যাটফর্ম স্পিকার হিসাবে নিজেদেরকে ইনভলভ করতে পেরেছে, সেইটার সম্বন্ধে আমাদের রাকা ম্যাডাম কিছু গাইডলাইন দেবেন। সো রাকা ম্যাডাম প্লিজ।
Thank you, Argha. Thanks to our honorable principal, Honorable Principal Madam Dr. Himadri Chakraborty Bhattacharya, our TCS Madam Professor Sarkar, our HOD Madam Dr. Urpita Sharnukar, Convened Dr. Argha Sarkar, and other departmental teachers and students in this seminar. It is a great pleasure to have an opportunity to share my thoughts with the students in this seminar, although I still consider myself as a humble student of life science. Student seminar is not only an opportunity for them to express themselves, but also allows them to participate in discussions which helps to clarify students' own views, allows them to participate in discussions which help to clarify students' own views, allows them to participate in arguments, debate, interactions, and over and above as an orator. It helps them to start thinking about new things which they think would be implemented practically. It helps to shy or reserved students find voice. The teacher can always take seminars to their students, but making students take seminars on the subject topics would always help them to learn the subject well. By taking seminars in front of their teachers and their friends, the students will able to talk before a crowd later without any difficulty. Also, they can learn mistakes, can improve their language. It enriches their knowledge, thought process, deduction ability, and communication skills. It will be the best and most effective way to study. So, seminars have an important role to play in the curriculum. In short, it is the best platform for the present students to face the world in future. I wish them a success. Thank you all.
Thank you so much, Radhavi. Really, the utility and the necessity of such like student seminar that is being organized by the Department of Botany, Bangabasi College today. So, student, I would like to share one more thing with all of you that this is the day 10th July. Exactly the 1 year before, the 11th July 2020, also a student seminar was held, and that was about biotechnology and its development in the current scenario. And on that day, two invited speakers were there. Our students like 4th SEM and 6th SEM, they were also the audiences there. In spite of that, various students from outside colleges, various teachers from outside colleges were present there. And that was a very, very memorable time. And exactly the 1 year later, we are organizing this type of seminar where no other invited speakers are there, as Principal Madam has also coined the same term. Exactly now, the invited speakers are our most beloved students that you are. So we are going to witness the event of student seminar. Please don't be bothered about anything. Just be frank and be vocal for your presentation. Be courageous and don't be upset, don't be anxious, don't fumble, as HOD Madam already said. Just be confident and deliver your presentation at your will. So just starting before the presentation, I would like to request our respected TCS Madam Professor Pushpita Sarkar to say a few words. So Pushpita Madam, please.
Very good afternoon, everybody. With the permission of our respected principal, Madam Dr. Himadri Chakraborty Bhattacharya, I just want to say just a few words because the main focus today is on the students, their seminar. It's not us. So I would say I'm really grateful. I'm really honored to be present in this seminar for the students, and I am really thankful to the Department of Botany, HOD, Dr. Orpita Sharnokar, Argha Sarkar, Oli Khan, Raka Dasgupta, Biplab Bakchi, every one of them, Devosri Gupta. I'm really very, very thankful because in every seminar they call me, and I am really honored, and I feel I'm enriching myself with their various topics that they discussed before. And today also, I'm looking forward to their seminar to the students. And for the students, I'm telling you, I'm really honored to be here because I want to see your presentation. I want to enrich myself. Thank you so much. Thank you so much.
Thank you so much, Nandini. Thank you so much for your impacting presence, and exactly your such presence encourage us to go forward. And in every seminar, in every events of the Department of Botany, you as being a guide, always been with us. So thank you, thanks to all of you. So seeking permission from all of the respected teachers and principals, and I would just forward the event exactly. Now we are with the time. And firstly, we will start the session of oral presentation, of PowerPoint presentation. Firstly, I would like to share some more things that few of students, those who have been already selected in this event, but due to their health issues or of yours, themselves or their parents or their relatives, and even for their heavy, not avoiding unavoidable some circumstances that they have been facing, and for that reason they are not with us to present themselves. But we assured and we wish that really surely we will get them to deliver their presentations, to deliver their work, to deliver the studies in future, surely. So now we start our session, and our first oral presentation that would be started by our beloved student of same sex, Odrija Khasnovish. And I would, one more thing I have to say, that in case of many students, they are presenting from their Android mobile. Very naturally in undergraduate condition. So when they are at undergraduate, exactly like us, when we have no computers, no laptops in our household, and that is not a crime. This is a very natural phenomenon. Still, they are using their mobile Android, and they have really did a hard work. Even they have did all the presentations by themselves. So exactly kudos to them for this type of hard work and labor that they have already did it. So I would present many of works for on behalf of them. They will just they are just upload their video. One more thing, Torun sir is joining. That is also very joyful to us. The Dr. Torun Kumar Sharma, Associate Professor in Botany and ex-head of the Department of Botany, he has joined us. So Torun sir, I think you are hearing me, and already Principal Madam is there, our TCS Pushpita Madam is there, HOD Madam is there, and all the other teachers are there of a department, and we are going to start our presentation session, seeking also your permission. So now I would request Odrija, please turn on your video, and I would present your topic so that you can deliver at yourself. Odrija, yes. So Odrija, firstly, introduce yourself.
Okay, so I am Odrija Khasnovish from Botany Department 6M.
Good. Just wait for your presentation. Odrija, is it visible?
Yes, sir.
So now this is your slide. So you may start, please.
Okay,
Just a minute to start your. All of students are requested to complete their total schedule only within 5 minutes. Their presentation will have to be completed within 5 minutes, as because we have some time constraint and so many participants are there. So 5 minutes for the presentation and 1 or 2 minutes for interaction. Okay, and I will manage all the time limits. I will just hammer up you at the exact 5th minute, and then you will stop your presentation. I think you will all be very much easier to deliver your presentation within 5 minutes. So Arjuna, please start.
Okay, so first of all, good evening to respected principal and to all of my respected teachers, juniors, and all my classmates of Botany Department. I am extremely grateful to get a chance to participate on this event, Student Seminar 2021. Am I audible?
Yeah, clearly audible. Please.
Okay. So firstly, I would like to start with a quotation of a famous writer, Sir Terror Tempest William, who told that cancer is not a disease of an individual, it is a disease of a family. Yes, it is a terror of all of us, and as the medicine is yet unknown to us, there are many methods to treat cancer. Still, there is not any, there is not any ultimate method with what we can treat cancer in a quick manner. So I would like to start. Can I proceed?
Yeah, sure.
Okay, so firstly, what is cancer? Cancer starts or initiates from a single cell. It is a disease of cells. When the cell grows or multiplies in a rapid manner, it grows or forms a tumor. It can be of two types, benign and malignant tumor. So benign tumors are grow mainly in a slow manner and harmful. But in case of malignant tumors, they grow rapidly, invade other cells, and they are actually harmful for other cells. Thus, a cancer cell forms. Now, I would like to go to the next slide. So, the causes and symptoms of cancer. Causes are actually the carcinogens, which are the causes of cancers. Some common carcinogens are alcohol, asbestos, formaldehyde, and some brown foods, and etc. And here I have showed the picture, and also in case of symptom, I have showed the picture of breast cancer. In case of India, every 4 minutes, a single individual woman is stressed with breast cancer. Other symptoms of cancers are fever, pain, etc. And also the anemia and uh, secretion of blood is also a uh, symptom of cancer. So on the next slide, yes, there is types of cancer. According to National Cancer Institute, there are A to Z types of cancers can be traced, but exceptional are Z, X, and Y. So now some common cancer types are thyroid cancer, breast cancer, colon cancer, etc. And cancer can be categorized into some broad categories like sarcoma, carcinoma, myeloma, lymphoma, leukemia, and central nervous system cancer. So now I will like to move on the cancer treatment. History of cancer is quite interesting. It was initiated in Egypt as in 1500 BCE, that is Common Era. Then from 1930, the cancer treatment was started with the method of radiation radiation therapy or with chemotherapy. Sorry. So now in the modern era, the cancer treatments are like biomarker treatment, hormone therapy, immunotherapy. So biomarkers are or tumor markers are targeted the proteins or genes which cause cancer, and in hormone therapy, mainly in case of prostate and breast cancer, hormone therapy is applicable. And then immunotherapy, their immunocells are targeted, and there are also some methods like radiation therapy, chemotherapy, which kills and shrinks, which helps to shrink the tumors and kills the cancer cells. Then in case of surgery, cancer cells are removed from the body. Now in the next slide, I would like to tell about some researches at 2020 based on cancer. Uh, Dr. Elvira Osalo has invented a new method that the coat on the cancer cell, which are mainly made up of proteins, this can be blocked, and when it can be blocked, the cancer cells also blocked by they stopped growing. Uh, uh, other than this, when in case of childhood brain tumor cases, immunotherapy plus TNF can be helped to reduce the brain tumor. So in the conclusion part, there are some other facts on cancer treatments. 4th February is celebrated as World Cancer Day, and some cancer is really tough to take or to
get. Your time is up. Just conclude.
Okay, sir. So every day in India, 1,300 people are dying out of cancer. But we also can say in cancer there is can, so we also can beat cancer in further future. Thank you.
So Trija, now the session is open for the interaction. Any participant, if you have any questions for Trija, you can do.
Adrija, what is the role of oncogenes in cancer?
Yes, ma'am. So, ma'am, oncogenes are mainly those genes which causes cancer.
How? Can you tell me? Do you know it?
By the effect of carcinogens, they rapidly divide and thus forms tumors. And when it is in malignant stage, it forms cancer.
The oncogenes actually get activated. Okay, they are in the dormant form, but they get activated when they come in contact with the carcinogen. Okay,
Yes,
there
yes,
is
ma'am.
gene amplification, then there is doubling of genes, this oncogenes, there is other factors there are, so that activate the oncogenes. You should.
Adrija, can you hear me?
Yes, ma'am.
A small question. See, you have very nicely described everything about cancer. Now, অলদো ক্যান্সার এটা অনেক দিন ধরে হচ্ছে, কিন্তু অনেক রকম ট্রিটমেন্টও বেরিয়েছে, সেগুলো তুমি একটু একটু টাচ করেছো ডেফিনেটলি। তাও কেন এই রোগটা বেড়ে যাচ্ছে, হালে এত বাড়ছে কেন? হোয়াট ইজ দ্য রিজন ফর ইট? সিম্পল রিজন।
Because we are not aware of cancer, of the common factors or common carcinogens. We are using it in a repeated manner.
Okay. And our lifestyle pattern also, taking junk foods, isn't it?
Yes,
Pollution,
ma'am.
exposed to pollutions. Okay, thank you.
Thank you, ma'am.
Thank you, Adrisha.
Thank you, sir.
Okay. In this context, I also want to know that I just forgot that our two teachers, Dr. Olika Hussain and Dr. Debashree Gupta, they are the two evaluators, and they are evaluating all the presentations. So we'll go for the next presentation.
Okay, sir, can I turn off the video and audio?
Sure.
Okay, sir.
So next turn is Anindita Ghosh of SEM 2. Anindita.
Yes, sir.
Anindita, turn on your video. Yes. Can you hear me clearly?
Yes, sir.
Okay. Then I am presenting your topic. Anindita, is it visible?
No, sir.
No visible.
No.
Anyone?
Yes, sir. Yes, sir.
Now it's visible, na?
Yes.
Okay. So please do continue. Start your presentation.
I am Anindita Ghosh from Botany Honors SEM 2 from Bangabasi College. Now I am present the topic of cyanobacteria. At first, cyanobacteria is the largest group of photosynthetic prokaryotes. We are also known as blue-green algae. It is consists of 150 genera and about 2,500 species. This is the thallus division of blue-green algae in two types of thallus division. One unicellular form. In this forms, the cells may be oval and spherical. The example of Glycocapta, Crucoccus, etc. Next, the colonies may be of two types: non-filamentous type, spherical, cubical, or filamentous type. In filamentous type, the example of Osteolia chitonema. The cell structure of cyanobacteria. Mainly the cell wall consists of 4 layers. It is composed of mucopeptide. Now the cell is also the algae is have a heterocyst formation. These are specialized cells of the filament distinguished from others by their thick wall, polar nodules, and homogeneous contents. It is commonly found in stichnometals and nostocyls. Some scientists considered it as the storehouse of food materials. Smith in 1951 pointed out it as vestigial spore, but according to Fay 1968, it is the site of nitrogen fiction. The reproduction of cyanobacteria. The cyanobacteria produces mainly vegetative and asexual reproduction. Sexual reproduction is now completely absent. The vegetative reproduction performs through fission, Synechococcus, fragmentation, Oskitoria, Hormogonia formation, nosto. During asexual reproduction, various types of asexual spores are formed. These are actinates, Anabaena sphaerica, and endospores, Dermocarpa, exospores, the example, Chamaecyphone. The economic importance of cyanobacteria. Harmful activities. Some members like microcystis anabaena from water bloom and can grow well in oxygen deficient water. Continuous respiration by submerged plants and animals during night time causes the depletion of oxygen to almost zero level. At that condition, mortality of both animals and other submerged plants takes place due to suffocation. Next, beneficial activities. Some algae like nostoc, anabaena, Chitonema can fix atmospheric nitrogen and form nitrogenous compounds. These compounds are further absorbed by the plants for their metabolic activity and increase yield. But certain non-heterocytous members like Plactonema boreanum are able to fix atmospheric nitrogen in anabolic condition. 2. Nasturtium commune is boiled and used as soap in China. And few species of Nostoc, Anabaena, Chitonema form a thick substratum over the soil, resulting in a reclamation of land. And I will respectfully thanksgiving all the teachers of my college and especially my mentor Dr. Oli Khan Shen and respectfully thanksgiving Dr. Argha Sarkar sir. Thank you, sir.
Anindita.
Hi, ma'am.
Yes, nice presentation. Now, this group is also known as blue-green algae, isn't it? Cyanobacteria.
Yes, ma'am.
তাহলে cyanobacteria, why it is called cyanobacteria? What is the reason behind it? Very simple reason.
Because the pigments, because their pigments are chlorophyll A and C phycocyanin, so it is called cyanobacteria. Because the pigment chlorophyll A pigments colored are green and C phycocyanin colored are blue. And some resembles of bacteria.
Yes, you are now coming to the point. Some characters resemble the bacteria. Do you remember one or two characters? No need to stress. If you remember, speak, or otherwise leave it.
No, ma'am.
Okay, okay, okay. Okay, thank you.
Devashree, have you any question? So anyone have any question regarding cyanobacteria presentation of Anindita? So thank you so much, Anindita. Thanks for your good presentation. Hope you will present much more good than it is. This is a very starting period. This is very initiation period for you. Then as a starter, it is a very good. Go on.
Okay, sir. Can I turn off my video and audio?
Sure.
Okay, sir.
So now the next presenter, Anisha Parveen of same to Botany Honors. So Anisha, now the platform is yours.
Good evening, sir.
Yeah, Anisha, please do continue. Now I will present your topic.
Hi, I'm Anisha Parveen from Botany Honors, SEM 2.
Is this clear to you, the screen, Anisha?
Yes, sir. Yes, sir.
Okay, so do continue. Proceed.
Next slide. Role of bryophyte in plant succession. Plant succession is also dependent on plants and habitat, and those matters are also interrelatable. Next slide, sir. Types of succession. Primary succession on the rock. Each succession proceeds from a primary base area or peritoneum substratum which has not been changed physically by organism. এই যে প্ল্যান্ট সাকসেশনটা হচ্ছে এটা বেয়ার রক থেকে স্টার্ট হয়, আর এখানে যে লাইকেনের এস্টাবলিশ হয় সেটা হচ্ছে লাইকেনটা হলো অ্যাসোসিয়েশন বিটুইন অ্যালগি অ্যান্ড ফাঞ্জাই। নেক্সট স্লাইড, স্যার। This is a picture of primary succession. Next slide. Primary succession on inorganic particulate. এখানে প্রাইমারি সাকসেশন শুরু হওয়ার আগে পৃথিবীতে কোনো প্ল্যান্ট ছিল না, তাই তখন ইনঅর্গানিক পার্টিকুলেটের যেসব ইনঅর্গানিক পার্টিকুলেট মানে ইনঅর্গানিক যে ম্যাটেরিয়াল গুলো আগে ছিল সয়েল তখন প্রিপেয়ার হয়নি, তখন পুরোটাই স্টোন ছিল। সেই ইনঅর্গানিক পার্টিকুলেট ভাঙার জন্য প্রথম যে সাকসেশন শুরু হয় সেটাই প্রাইমারি সাকসেশন অন ইনঅর্গানিক পার্টিকুলেট, অ্যান্ড দ্যাট ওয়াজ স্টার্টেড বাই অ্যালগি অ্যান্ড ফাঞ্জাই ইন ইলাবোরেটিভ ম্যানার। প্রাইমারি সাকসেশন অন ওয়াটার। এখানে যে প্রাইমারি সাকসেশন হচ্ছে গাছে, এখানে সাকসেশনের জন্য স্যার কোনো রকম জল থাকে না, এখানে ড্রাই অবস্থাতেই প্ল্যান্টের সাকসেশন হয়। অটো সাকসেশন যেটা হচ্ছে, অটো সাকসেশনে একটা ফরেস্টের ওপর ডিপেন্ড করে হচ্ছে, সেই ফরেস্টে নানান প্রাকৃতিক বিপর্যয় লাইক ফরেস্ট ফায়ার, হারিকেন, হারভেস্টিং, ওয়াইন ফ্লু ইত্যাদি হওয়ার ফলে যে ক্ষতি হচ্ছে, সেগুলো হওয়ার ফলে আবার সেখানে যে গাছ মানে আবার যে সাকসেশনটা স্টার্ট হচ্ছে, এটা হচ্ছে অটো সাকসেশন। নেক্সট স্লাইড, স্যার। This is a picture of primary succession in water. Next slide. Secondary succession. It is the succession that occupies the secondary area previously colonized. এই সাকসেশনটা যেখানে স্টার্ট হয় সেখানে আগে থেকেই সাকসেশন ছিল, কিন্তু কিছু প্রাকৃতিক বিপর্যয় লাইক আজ ফরেস্ট ফায়ার, হারিকেন, হারভেস্টিং এসব হওয়ার ফলে সেখানে ওই সাকসেশনটা নষ্ট হয়ে গিয়েছিল বা ডিস্ট্রাক্টেড হয়ে গিয়েছিল, এবং এটা হওয়ার অনেকদিন পরে আমরা আবার হয়তো সেখানে প্ল্যান্ট এস্টাবলিশ হতে দেখেছি বা প্ল্যান্ট গ্রো হতে দেখেছি। নেক্সট স্লাইড, স্যার। Cyclic succession. This cyclic succession model was presented by Alexander Ward in 1947. Suppose একটা বড় জঙ্গলে এই সাকসেশনটা হচ্ছে, সেখানে নানান বিপর্যয়, যেমন ওখানে অনেক পশু পাখি থাকার কারণে হয়তো গাছপালা নষ্ট হয়ে যাচ্ছে, কিন্তু গাছপালা নষ্ট হয়ে যাওয়ার ফলেও অনেকদিন পর আবার ওখানে যে গাছপালা তৈরি হচ্ছে, প্ল্যান্ট গ্রো করছে, প্ল্যান্ট এস্টাবলিশ হচ্ছে, এটা cyclic succession. And succession and struggles, both primary and secondary succession are normally marked by a progressive increase in species diversity. এখানে প্রাইমারি সাকসেশন এবং সেকেন্ডারি সাকসেশন, বোথ সাকসেশনটাই এখানে ডিসক্রাইব করা হয়েছে। নেক্সট স্লাইড স্যার। Ecological succession. Ecological succession is the process that describes how the structure of biological community, that is an interacting group of various species, desert, forest, grassland, marine environment, and so on, changes over time.
Anusha,
Yes, sir.
have you anything to say?
No, sir.
So are you completing? You're completing?
Yes, sir.
So just say that you are completing your topic.
Anisha,
Yes, sir.
what is pioneer of climax communication? Do you know climax?
No. No, sir.
You should know the climax because you are thinking about the succession. The succession is the climax. There is a very important climax. Okay.
Anisha,
Yes, sir.
one basic question is this: what is succession? কি বোঝো তুমি succession বলতে কি বোঝো?
সেটাই
স্যার সাকসেশন হচ্ছে একটা মেইন পার্ট যেটা দ্বারা আমরা প্ল্যান্টকে ভাগ করে থাকি। মানে যখন পৃথিবীতে প্ল্যান্ট ছিল না তখন এই সাকসেশন জিনিসটা স্টার্ট হয়েছিল। কি বললেন?
কি সেটা সাকসেশনটা কি? সাকসেশন স্টার্ট হয়েছিল তো বুঝলাম, কিন্তু জিনিসটা কি কি জানো?
নো স্যার।
না, তোমার তো টোটাল টপিকটাই প্রাইমারি সাকসেশন, সেকেন্ডারি সাকসেশন, এগুলো বিভিন্ন টাইপস বললে, রোল অফ ব্রায়োফাইটস বললে, কিন্তু হোয়াট ইজ সাকসেশন তুমি জানো না, তাই হয় কি করে? নিশ্চয়ই তুমি জানো। আমার মনে হয় তুমি টেনশন করছো। টেনশন না করে ফ্রিলি বলো একদম, যে সাকসেশন বলতে আমি কি বুঝি, তোমার কি মনে হয়, একদম নিজের ধারণা পরিষ্কার করে বলো। তুমি তো এটা পড়েছো, প্রাইমারি সাকসেশন, সেকেন্ডারি সাকসেশন পড়েছো, এই তো এগুলো বললে এক্ষুনি, ভালোই বলেছো। এবার বলো সাকসেশনটা কি, জিনিসটা কি?
সাকসেশন যখন স্টার্ট হয়েছিল তখন তো প্ল্যান্টস ছিল না পৃথিবীতে।
সাকসেশন যখন স্টার্ট হয়েছিল তখন প্ল্যান্ট ছিল না, তো কারা ছিল? শোনো, আচ্ছা ঠিক আছে, তোমার একটু কনফিউজড হয়ে গেছো, কোনো অসুবিধার কিছু নেই, তোমার কিছু শরীরটাও মনে হচ্ছে ঠিক নেই।
হ্যাঁ স্যার।
হ্যাঁ আচ্ছা, শুনে নাও, যে কোনো টপিক তুমি যখন বলবে তার বেসিক জিনিসগুলো সবসময় মাথায় রাখতে হবে, তুমি কি বলছো।
না অনিশা,
হ্যাঁ স্যার,
আমি একটা কথা বলছি, তুমি যে টপিকটা বললে ভালো বলেছো, ভালো লিখেছো, কিন্তু তুমি ওটাকে হৃদয়ঙ্গম করতে পারোনি, তুমি ওটাকে বুঝতে পারোনি। হ্যাঁ, সাকসেসের ব্যাপারটাকে তোমাকে বুঝতে হবে। তুমি একটা বই থেকে লিখে দিলে সেটা কোনো বড় কথা নয়, ঠিক আছে? তুমি এটাকে বোঝার চেষ্টা করো, হোয়াট ইজ সাকসেশন, যেটা অর্ঘ স্যার বললে হোয়াট ইজ সাকসেশন। হ্যাঁ, এই জিনিসটাকে বোঝার চেষ্টা করো, ঠিক আছে? ওকে, তবে তুমি ভালো বলেছো।
ঠিক আছে, তোমার ডেলিভারেশন ভালোই ছিল, ভালোই করেছো। নেক্সট টাইম যেগুলো স্যারেরা বললেন ওগুলো আরো ভালো করে করবে। ওকে
ওকে,
থ্যাংক ইউ।
যে কোনো টপিক যখন তুই চয়েস করবি না, তখনই দেখে নিবি যে এক্স্যাক্টলি সেই টপিকটা আমি কতটা নিজে পারি বা নিজে জানি। একটু কনফিডেন্স থাকলে সেটাই চুজ করবি, ঠিক আছে? বলাটা যেমন ভালো হয়েছে, তার সাথে যে কেউ প্রশ্ন করতেই পারে, যে একদম বেসিক কিছু প্রশ্ন। তাই সাকসেশন কি, যদি তুই প্রশ্ন করছি না, উত্তরটাও বলছি তোকে সহজ ভাষায়। সাকসেশন মানে হলো একজনকে সরিয়ে সেই জায়গায় আরেকজনের আসা, একজনের জায়গাটাকে আরেকজন অধিকার করে, এটা হচ্ছে সাকসেশনের প্লেন মানে। এই যে ব্রায়োফাইটস, তার মানে ব্রায়োফাইটস এমন একটা হ্যাবিট, প্ল্যান্ট হ্যাবিট, যে ব্রায়োফাইট তারা বলছে আর্লি কলোনাইজার এদেরকে বলা হয়। মানে এরাই আর কি প্রথম উপনিবেশ গড়ে তুলেছিল কোনো একটা জায়গাতে, আবার এদের সাকসেশনের জন্য এদের ভূমিকাটা এই কারণেই, কারণ এরা যেখানে থাকবে সেখানে নিজেদের এতটা দল বাড়িয়ে তুলবে, নিজেদের এমন বড় উপনিবেশ গড়ে তুলবে যে সেই জায়গাটা যেন তাদেরই হয়ে যায়। কিন্তু এই পৃথিবীর নিয়মই হচ্ছে কেউ কোথাও স্থায়ী নয়, সেই কারণেই খুব স্বাভাবিকভাবেই ব্রায়োফাইটস তার নিজের জায়গা থেকে সরে যাবে, আর একজনকে সেখানে ঠাঁই দেবে, কিন্তু সেই জায়গাটা তাকে সুইটেবল করে দেবে আরেকজনের জন্য, যাতে সেখানে এসে সে থাকতে পারে। তখনই সেই সাকসেশনটা এস্টাবলিশ হয়, তখনই সেই সাকসেশনটা সাকসেসফুল হয়। এবার তো প্রাইমারি সেকেন্ডারি তার ভাগ, সেগুলো তো অন্য জিনিস। বুঝতে পারছিস? আমি একদম সহজ ভাষায় কিছু বলার চেষ্টা করলাম।
ইয়েস স্যার।
ঠিক আছে, ওকে, থ্যাংক ইউ অনিশা। আমাদের নেক্সট প্রেজেন্টেশনে আমরা চলে যাব। অনিশা, এবার তোমার ভিডিও এবং অডিও অফ করতে পারো। আমাদের নেক্সট প্রেজেন্টেশন, প্রেজেন্টার অঞ্জলি শাও, সেম 6। Anjali?
Yes, sir.
Can you hear me clearly?
Yes, sir.
Oh, just wait. I am presenting the topic. Is it visible, Anjali?
Yes, sir.
Okay. Start and say थोड़ा सा louder बोलना, हाँ?
ठीक है.
ठीक, चलो start करो.
Good evening, everyone, and myself Anjali Shah, and I am here to present an introduction to mineral nutrition in plants. Sir, next slide. As we know, all organisms draw materials from the environment in order to maintain their growth, metabolism, and development. This plant nutrition is treated as a two-step topic, that is organic nutrition and inorganic nutrition. Organic nutrition mainly focuses on that absorption and metabolism of carbon, hydrogen, oxygen, nitrogen. And since these elements are available from atmosphere and water, so this constitutes non-mineral nutrition. Whereas plants fulfill their inorganic constituent or inorganic nutrition chiefly through soil. And this inorganic nutrition mainly consists of minerals, so it is known as mineral nutrition. So if I need to define what is mineral nutrition, it is the absorption and metabolism of various inorganic substances, mainly minerals, which are used by the plant for their growth and metabolism. Now, how we can study the mineral requirements of plant. In 1860, German botanist Julius von Sass demonstrated for the first time that a plant can be grown in completely absence of soil. There is no need of any soil. So this growth of plant in a nutrient medium is considered as hydroponics or soilless culture. In this experiment, the plant roots is dipped in the nutrient solution, and that elements which are required for their growth are supplied from outside in varied concentrations, so that a suitable nutrient solution is formed for the plant to grow properly. And with this hydroponic culture, we can know many things like what are the essentiality of mineral nutrients, and what are their deficiency symptoms if they are in less amount, and what are the toxic effects if it is slightly more in concentration, and the possible interaction among the different elements present in the plants. Now, what are essential elements? So essential mineral elements are those elements which are required by the plant directly for their growth and metabolism, and without which their life cycle is incomplete. And if it is present in less amount, deficiency symptom can be seen. And according to Arnon, a scientist, botanist, according to Arnon, the criteria of essentiality of mineral nutrients were that the element should be directly involved in the growth and metabolism of the plant, and it should be irreplaceable. It cannot be replaced by any other element, and it is necessary for normal growth and reproduction of plants. Arnon also classified these mineral elements in two different parts according to the requirements of the plants, like the macronutrients and micronutrients. Macronutrients are the one which are needed in large amount by the plant cell. Mostly it must be 1 to 10 microgram per liter of dry matter, and microelements are comparatively required in less amount, that is its concentration should be 0.1 to 1.0 microgram per liter of dry matter. And this microelement, if it is present in slight excess amount, it does not cause any toxic effect to the plants. Whereas microelements, if it is present in slight more concentration, it can show toxic effects to the plants. So next slide. The macroelements include nitrogen, phosphorus, potassium, calcium, magnesium, carbon, oxygen, hydrogen. And the micronutrients include iron, boron, chlorine, manganese, zinc, copper, molybdenum, nickel. So here in this table, we can see there are various elements here and their role in the plants. So you see carbon it is used in photosynthesis, the constituent of carbohydrates. Hydrogen maintains the osmotic balance. Oxygen is required for respiration. Then calcium, it is a component of cell wall. Boron, believed to be important in sugar translocation, carbohydrate metabolism. Iron, it is involved in chlorophyll synthesis and enzymes for electron transfer. Magnesium, it is an enzyme activator and is a component of chlorophyll. Nickel, it is used in proper functioning of the enzyme and is found necessary in seed germination. Zinc is also involved in enzyme system and they regulate various metabolic activities. So, now the elements which are present in less concentration show some deficient conditions. So there is a particular critical concentration below which if an element is present, it is known to be deficient. Critical concentration is that concentration of an element below which the plant growth is retarded. So here are some deficient symptoms. So associated with the plants, with nitrogen, potassium, magnesium, sulfur.
Anjali, is it timer up or just conclude the matter?
Okay, sir. So he has some deficiency symptoms associated with the plants which are involved, which are associated with various deficient minerals like chlorosis, purple coloration, the dieback disease, wilted leaves, whiptail disease of the leaf. And that's all for today. Thank you very much. Thank you all the teachers and our principals. Thank you.
Thank you, Anjali. Now the interaction session.
Can I ask a question or no?
Yeah, sure, Lily.
This is Anjali. This is from a layman's point of view. Okay, what is the most useful nutrition for flowering plants? Organic or inorganic or a mixture of both? I just want to know, just for my sake.
Flowering plants. It needs to flower from vegetative to
Yeah, flowering. Yes, flowering, growing flowers, flowering plant.
I think inorganic nutrition.
What is it composed of? Inorganic?
Inorganic nutrition are those that minerals present in the soil that plants derive it from the soil. That nickel, molybdenum, manganese, zinc, and all other 16 to 17 elements are there which are considered to be essential elements, and which are 3 to 4 are consumed from atmosphere and water, but others are consumed from soil in the form of minerals.
Anjali,
okay.
Very good question. Asking Pushpita Di. It's a very important question. You should satisfy her.
Anjali, I need to ask one question. I just noticed in one of your slides that non-essential elements are not required by plants, but they are present in plant. Can you justify this? Your presentation was very good, I must say.
Thank you, ma'am. Actually, these elements are present in the plant, but they are not directly involved in a plant's metabolism or plant's growth. They may act as a beneficial element that they can be.
They have some utility. So they have some utility.
Yes, ma'am. Yes, ma'am.
So you cannot straight away say that they are not required by plant. Can you say that? I don't know.
No, ma'am. Sorry.
Okay.
Thank you, Anjali. That was a very good presentation.
Yes, the presentation was very good.
Thank you.
Thank you so much.
Thanks, Anjali. It was very nice presentation and the interaction session was also good. I think you have already got some visions from the questions of the sir and madam.
Yes, sir.
Okay, very good.
Thank you.
So So our next presentation. Now it's time for Anisha Banerjee of Sem 4.
Hello, sir. Am I audible?
Yeah, clearly audible. Yes, Anisha.
Sir, can I start now?
Yeah, I will present your topic.
Okay. So, good evening to all my.
Is it visible clearly, Anisha?
Yes, sir. Yes, sir. It is visible.
Okay, to proceed.
Sir. Good evening to respected principal ma'am, to all my respected teachers, seniors, juniors, and my fellow classmates. Myself, Anisha Banerjee, a student of semester 4, Botany Honors. And for today's presentation, my topic is nutritional and medicinal value of mushrooms. So to start with the presentation, let me say that we all are aware that mushrooms are used worldwide as food in several parts of the world. But while savoring the delicacies made by mushroom, we often forget to ponder upon its values like medicinal and nutritional values. So in my today's presentation, I'll deal with the medicinal and nutritional and other values of mushroom. Sir, no, we'll have to just skip 2 slides.
Okay.
No, sir, you have to go, from the beginning, after the first slide. Yes, sir, this one. So this slide deals with basically the enzyme inhibiting activity of mushroom. Let us see what impact mushroom has as an enzyme inhibitor. Mushrooms are known to inhibit the production of enzyme aromatase, which reduces the risk of breast cancer in women. Mushrooms are widely used as nutraceuticals, which are taken as capsules and tablets. Pleurotas produce industrial enzymes like xylanases, pectinases, and cellulases. Now, coming to the next point, we will see the nutritional value of mushrooms. Uh, uh, During the ancient time, Romans and Greeks referred mushroom by the food for God because they provide sufficient bodybuilding nutrients and they also play a noticeable role in the prevention of diseases. Moving on to the next slide, we will see in more details how mushrooms play a very much important role in protein content. For example, uh, Agaricus bisporus, uh, it has the highest level of riboflavin, vitamin, and to look into more details, if we see that mushroom has twice as more nutrition and cabbage than asparagus. Now, carbohydrate content of mushroom. Carbohydrate content in mushroom is very minimal. That is why they are very effective in keto diet. And for those people who tend to maintain low weight, it is very essential as it contains very lower amount of calorie. Now, coming to the next slide, we will see the mineral content in mushroom. Mineral content in mushroom. Mineral content in mushroom is very much abundant. We will see that molybdenum, magnesium, and other minerals are present in very good amount in mushroom. Example of such mushroom where the minerals are present in very much abundance are agaricus, ganoderma, and many others. Next slide. Now, in this slide, we will see mushroom plays a very important role as dietary fiber. Mushroom contain chitin, cellulase, and many polysaccharides, which are indigestible in humans, but which are indigestible in humans, thus they are used as roughage. And we all know from a very beginning of our life that what role roughage plays in our life. Roughage is very much essential in our diet chart because it helps the bowel movement to, it normalizes bowel movement so that constipation doesn't occur. And many other, it controls the blood sugar level. They lower the density of lipoprotein and many. Next slide. Now we will see the medicinal value of mushroom. As I already mentioned that mushrooms are used as nutraceuticals What are exactly nutraceuticals? Nutraceuticals are nothing but these are the extracts of mycelia or the fruiting bodies of mushrooms, which are manufactured in markets like in form of tablets and capsules. Mushroom. Now we'll see in more details how these mushrooms are actually helping to cope up medical issues. First point deals with hematological effect. What does hematological effect of mushroom concerns with? Scientists have seen that mushroom consumption helps in platelet therapy. Examples of such mushrooms are Lentinula idoides, Volviriella volvicea, Ganoderma lucidum, etc. Next point is antiviral effects. Antiviral effects deals with it is becoming widely known that the extracts from mushrooms have been shown to display antibacterial and anti-inflammatory effects. For example, phenolic fraction of the mushroom extract capable of conferring an antiviral activity. The example of this mushroom is Lentinula idoides. The fruiting body of this mushroom is seen to cure the influenza virus. Next, we are coming to carcinogenity of mushroom. Yes, sir.
Just conclude.
Okay, sir. Next 2 points are carcinogenicity and nutraceuticals. Carcinogenicity is nothing but the chronic effect of mushrooms. It's not that mushroom doesn't happen to have any toxicity. There is toxicity in mushroom, but in a very less amount. For example, in Aspergillus flavus, it contains an alpha-toxin B, which is a harmful toxin. Now, last slide, we are coming to the conclusion, where it is said that mushrooms helps in various amount of ways, like increasing, like helping the patients who have undergone the chemotherapy or radiotherapy by reducing the side effects, then inducing tumor regression, stopping hair loss, and many others. Thank you.
Thank you, Anisha. I think the timing custom animation is not working and it's still showing. That's why it is being disturbed, Anisha. Okay, not so problematic. The presentation was good. Now it's open for the interaction.
One question, Anisha, your
Yes,
presentation was very good. A very simple question. You have covered nutraceutical and everything.
Yes, ma'am.
But do you know which country is the largest producer of mushrooms, especially edible and medicinal? Have you any idea? A very simple.
No, ma'am. I'm sorry, I don't know this answer.
It's China, next Japan, Taiwan. China is the top leading. And they do a lot of work in medicinal mushrooms, nutraceuticals, etc. Okay, thank you.
Okay, ma'am.
Anisha,
Yes, sir.
you have just mentioned the agaricus and Volvarella, but
yes, sir.
there are Morchella, Pilotus, so many whatever it is, so many fungus, edible fungus, they are also very important. You should mention the Pilotus and Morchella. Okay?
Sir, I mentioned the name of Theodorus.
Okay.
Yes, sir.
So, Prisha, thank you so much for your nice presentation, and also the interaction session was good. Thank you.
Thank you, sir.
Now the next one. It's turn for Arundhati Singho, option 6. Yes, Arundhati, can you hear me? Arundhati?
Unmute yourself.
Yes, sir.
Am I audible?
Yes, sir.
Okay, just wait. Is it clear?
Hello?
Is it clear?
Yes.
Okay, proceed.
Hello, good evening, everyone. Myself, Arundhati Sinha. Today, the topic of my presentation is all about caseation acid metabolism mechanism and its ecological significance. Now I am talking about it. Casugation acid metabolism is an photosynthetic specialization beyond C3 and C4 that permit the opening of the stomata aperture and net CO2 uptake at night. It was observed by the botanists Ranson and Thomas in the Casuraceae family of succulent. Next. The CAM occur in hundreds of species in 20 family of angiosperm. Some of these families are Liliaceae, Casuraceae, Euphorbiaceae, Cactaceae. CAM is an adaptation. CAM is an adaptation for increased efficiency in the use of water and so is typically found in plant growing in acidic condition. Now moving to the next slide. Mechanism of CAM. During night, the plant employing CAM, a plant employing CAM has been stomata open, allowing CO2 to enter and be fixed as organic acid by the PEP reaction, similar to the C4 pathway. The result, organic acid, are stored in vacuole for later use. As the Kelvin cycle cannot operate without ATP and NADP, product of light-dependent reaction that do not take place at night. In this image, we can see in this image. Slide. In this image, we can see during night CO2 enter to the plant cell and stored as organic acid in vacuole. Next slide, please. The next. Mechanism of CAM. During the day, stomata close to conserve water and the CO2 storing organic acid are released from the vacuole of the mesophyll cell. An enzyme of stoma of chloroplast release the CO2 which entered into the Kelvin cycle. In this image, we can see from organic acid CO2 release and entered to the Kelvin cycle. That's the pathway. Now I will discuss about some ecological significance of CAM. CAM plants are specially suited to the dry habitats such as in desert. These plants have remarkable capacity to attain high biomass under condition of high evaporation rate of scattered rainfall, which are otherwise insufficient for growing of crop plants. CAM plants have different comparative advantage over C3 and C4 plants in dry habitat. Now I will now the conclusion is now the acknowledgement. I want to express soulful thanks to Dr. Himadri Bhattacharya Chakraborty, Honorable Principal Dr. Arpita Sarkar, HOD Department of Botany, Bangabasi College, for providing me an opportunity to undertake this condition. Under this curriculum and this pandemic condition, I am also grateful to Dr. Argha Sarkar, Assistant Professor, Department of Botany, Bangabasi College, for constant helping me. And here the references are. Thank you.
থ্যাংক ইউ অরুন্ধতি। এত টেনশন করছিস কেন? টেক আউট কম্পল হচ্ছে। ঠিক আছে, ইন্টারেকশন সেশন ভালো হয়েছে, বাট একটু বেশি টেনশন করে ফেলছিস।
অরুন্ধতি, একটা কথা বলি, সেটা হচ্ছে যে স্লাইডস গুলো তোমার যে থাকে সেটাতে না তুমি মানে ওটা রিডিং পড়বে না। হ্যাঁ, এরপর থেকে যখন প্রেজেন্টেশন করবে তখন নিজে থেকে কিছুটা বলার চেষ্টা করবে। তোমরা এটা প্রথম প্রেজেন্টেশন করছো, ঠিক আছে, কিন্তু কি হয় অনেক সময় টেকনিক্যাল ইস্যুতে স্লাইডস এলো না, তোমার বলাও থেমে গেল, সেটা তো ঠিক নয়। পরের বারকে একটু চেষ্টা করো। ঠিক আছে, ভালো হয়েছে।
একদম অরুন্ধতি ম্যাডাম যেটা বললেন, সবসময় রিডিং করবো না, আমি কোনো কিছু আগে পড়ে রাখবো এবং সেটার সম্বন্ধে আমি সবাইকে জানাবো, তাতে আমার স্লাইড আসুক কি না আসুক আমার বলা কিন্তু কখনোই থামবে না। ঠিক আছে? আর সবসময় হুবহু লাইন বাই লাইন বলার চেষ্টা করবি না, নিজের মতো করে একটুখানি সাজিয়ে গুছিয়ে বল। ঠিক আছে? আরো ভালো হবে। স্টিল ইটস গুড। থ্যাংক ইউ অরুণিতা। Next participant, Devaroti Pal of SEM 6.
Yes, sir. Sir, am I audible?
Yeah, sure.
Sir, I shall present my screen.
Okay, very good. Do I am stop presenting. Yeah, it's presenting. Please open your PPT. Yeah, now go to the full screen mode. Yeah, exactly. This is start.
Okay, sir. Good evening to all of our respected dignitaries and fellow students. I am Devaroti Pal, student of Bangabasi College, 6th semester Botany Honours. I feel very blessed to participate in such an interesting seminar. Today I want to focus on the topic mineral nutrition in plants. When chemical compounds and elements necessary for plant growth, metabolism and other external activities are absorbed in plants is called nutrition, and the chemical compounds are called nutrients. So the mineral nutrition is the relationship between plants and all chemical present in the environment. Next is relationship between essential element and beneficial element. All essential elements are beneficial for plant, but beneficial elements are not essential for all the time in plants, because cobalt, sodium, selenium, silicon, etc. are beneficial element which is helpful for the plant growth, but in their absence plant growth doesn't hamper. In other words, in other hands, nitrogen, phosphorus, sulfur, etc. are the essential element and they are the building blocks of the plant body. So without them plant cannot survive. Next is the criteria for essentiality. As I have mentioned earlier that essential elements are the building blocks of the plant body. So they directly involved in plant physiological and metabolical activities. Another important point is essential elements should not be replaceable by any other molecules or elements. Next, I am discussing on essential elements, which is categories into two which is classified into two subcategories: micronutrients and macronutrients. Micronutrients is needed in tissue concentrations equal to or less than 100 microgram per gram of dry matter, whereas macronutrient is needed in tissue concentration equal to or more than 1000 microgram per gram of dry matter. Micronutrients are present in the plant in very small amount, whereas macronutrients are present in the plant in very detectable quantities. There are the 8 microelements present in the soil, such as nitrogen, copper, etc., whereas in macroelements the number of such element is 9, like carbon, hydrogen, oxygen, etc. Next is method of study and use of nutrient solution. As population of the world is increasing day by day, so we need to more reliable way to meet the demand of food. So one such technique is hydroponics. Hydroponics is a technique where plant grow in an aqueous media in an aqueous media without soil. Successfully in hydroponics, successfully growing crops are tomatoes, cucumber, Arabidopsis thaliana, which is the model plant of in our botany, and this phenomenon this experiment was first observed by Julius von Sachs in 19th century. Next, here I have made a chart containing essential elements, their roles, their deficiency symptoms and pictures. One of the most important essential element present in the soil is nitrogen, which is absorbed from soil to the plant by nitrate form. It plays an important role in the formation of DNA, RNA, proteins, that is amino acids. It's also the main component of chlorophyll cytochromes. Amino acid, that is nitrogen, which converts into vitamins, coenzymes, alkaloids, which helps for plant growth. Now come to the deficiency points. Chlorosis is in older leaves occur in deficiency of nitrogen. Any other deficiency symptoms are reduced plant growth, respiration is reduced, etc. Another important molecule is molybdenum, which activates the enzyme of nitrate reductase and reduces the nitrogen fixation. Whiptail of cauliflower, one of the most common disease in cauliflower, caused by the molybdenum of plant. Molybdenum. This is the picture of whiptail of cauliflower. Next is carbon, hydrogen, oxygen. This is the structural element of the plant. So without this structural element, plant cannot survive. So I have put here a normal healthy growing plant. And also hydrogen, that is H plus, maintain the pH. Oxygen also helps in respiration. Next is phosphorus. Next to the nitrogen, phosphorus is the most essential element present in the soil, which helps in DNA, RNA, and ATP, ATP formation. In phosphorus is also present in cell membrane as a phospholipids. It's also play a key role in oxidation, reduction, reaction, fat metabolism. Premature leaf fall, necrosis, protein synthesis in decreases. It is the quite common symptoms of phosphorus deficiency. Now boron, it helps in uptake of calcium and plays a key role in the sugar translocation and growth of pollen tube. In deficiency of boron, stem root dies and heart or rot beats may happen for the deficiency of boron. Manganese helps in the activation of chlorophyll and formation of indole acetic acid. Manganese also helps in photosynthesis of oxygen evolving complex. Mass spot of disease of pea is very common disease caused by manganese deficiency. Calcium is the main component of middle lamella present in between two cells, and calcium also helps in spindle formation during anaphase of mitotic cell division. So it is quite common that abnormalities in chromosome can be categorized in the deficiency of calcium. Chelating agents are those agents or molecule which required by plant for the stable of different biomolecule. That is, chelating agent bind with the metal ion to produce water soluble chemical compound. One such chelating agent is EDTA. Next, I come to the conclusion part. Plants mainly absorb nutrition or inorganic elements from soil. Out of 105 elements, there are 21 essential and beneficial elements helps in plant normal growth and development. Some prominent deficiency symptoms are necrosis, chlorosis, stunted growth, etc. Next, I must convey my sincere regard and gratitude to the principal of Bangabasi College. Yes, and then I also thankful to my mentor, Dr. Sarkar ma'am, and my family to being constant support towards the implementation of this presentation. These are the references, and thank you.
Debaruti, can you hear me?
Yes, ma'am.
You have said very well, and your presentation was very nice too. But you have talked about hydroponics. Can you tell something about very popular method now, that is aeroponics? Anything regarding that?
Aeroponics is a treatment, cultivation treatment where through air nutrients are flow through the room in the roots, and from the air plant can absorb the mineral nutrition, and this is the aeroponic system.
Devarati,
Yes, sir.
Your art and style of deliberation is very, very good. It is very fine. I have a small question. What is the fundamental role of iron and magnesium in the formation of building block of some parts of plants?
Yes, sir. Magnesium, which is the component of chlorophyll.
Yes.
So without magnesium, plant cannot survive.
And iron?
Iron is the main component for root growing structure, and it activates many enzymes which helps in plant development.
You would know that during the formation, at the first stage of the formation of chlorophyll, iron is needed, and after some time, iron is replaced by magnesium. Okay, thank you.
Yes, sir.
Very good presentation.
Nice presentation. Well done. Only a small question. This is not a question. Do we grow hydroponics in Bengal, West Bengal?
Yes, ma'am. It is cultivated in anywhere.
কি কিছু জানা আছে কি কি করা হয়?
Hydroponics, in hydroponics we can grow strawberry, lettuce, model plant. Our model plant Arabidopsis thaliana.
That is model plant. Okay, thank you.
দেবারতী একটা কথা বলি, আইআইটি খড়গপুরের যে এগ্রিকালচার ডিপার্টমেন্ট, সেখানে বড় একটা ফার্ম আছে, সেখানে হাইড্রোপনিক্সে প্রচুর কাল্টিভেশন হয়। হ্যাঁ, হাইড্রোপনিক্স হয়, তারপর এয়ারোপনিক্স হয়। ঠিক আছে, ওটা একটু জেনে রেখো।
ওকে ম্যাম।
Devarati, very good presentation. It's nice to see that your gesture is very good to say that deliberation in a very standing moment, like an offline presentation. It's good. One thing that you should always keep it in mind that when you are delivering, the layman that it's not seeing you or not seeing your presentation on behalf of you, he or she should not be understood about that you are in very hurry. Don't be so busy to present your topic. It may be the time constant at 5 minutes for your oral presentation. But your lecture should be so much arranged so that the 5 minutes time it cannot be such a clumsy condition to you so that you are being in a very hurried moment to complete your topic. এবং সেই কারণেই তুই যখনই কিছু বলতে যাচ্ছিস মনে হচ্ছিল খুব তাড়াহুড়োয় আছিস, স্টিল ধরবি আর কি। হ্যাঁ, এটা আরো আস্তে 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 করতে করতে প্র্যাকটিস করতে করতে হয়ে যাবে। স্টিল হ্যাঁ, হয়তো তোর মানে আমি তোকে থামাইনি একটাই কারণে, তার কারণ যখন তোর 5 মিনিটস কমপ্লিট হয়েছে সেই সময় তুই কনক্লুশনের স্লাইডটাই ছিলিস। এটা তোকে বলে দিলাম, তুই বুঝতে পারলি তোর টাইম ম্যানেজমেন্ট। ঠিক আছে, প্রত্যেকেই এটা বলার যে নিজেরা বাড়িতে বারবার প্র্যাকটিস করতে করতে এটা হয়, এটা একদিনে হয় না। স্টিল যতটা যে অবধি করেছো মানে খুব খুব ভালো, ফেয়ার এনাফ।
থ্যাংক ইউ
ঠিক আছে
স্যার, অ্যাকচুয়ালি টাইমিং এর জন্যই আমি একটু স্পিডে বলেছি।
হ্যাঁ, এবং সেই কারণেই আমাকে তাহলে আমার বলাটাকে এমনই করতে হবে বা আমি এমনই কিছু বলবো যেটা 5 মিনিটের মধ্যে শেষ হয়, এমন কিছু বেশি বলবো না যেটা 5 মিনিটে শেষ হবে না। ঠিক আছে, আর আবারও একটা কথা যেটা আমি এর আগের দিনও তোকে আলাদা করে বলেছিলাম, লাস্ট স্লাইড যেখানে রেফারেন্স গুলো মেনশন করা আছে, all the references are very good but there is no any reference of undergraduate study textbook. তোমার হাইদেন ভট্টাচার্যের বই ওখানে থাকার কথা, বা আন্ডার গ্রাজুয়েটের যে রেফারেন্স গুলো সেগুলো অবশ্যই সেখানে একটা বা দুটো তুমি মেনশন করবে আদার দ্যান দ্য রেফারেন্সেস। তাহলে তোমার বোঝা যাবে যে এক্স্যাক্টলি আমি আন্ডার গ্রাজুয়েটের এই বইগুলো ফলো করেছি, বোঝা করছো। ঠিক আছে, আদারওয়াইজ প্রেজেন্টেশন ইজ গুড, স্লাইডস আর ভেরি গুড। থ্যাংক ইউ।
May I turn my video off?
Yeah, sure. Now the next presentation, another member of SEM 6, Goldie Pandey of SEM 6.
Yes, sir. Good afternoon, sir.
Good afternoon, Goldie. We are not. Yeah, your video is now clear. Do you hear me?
Yes, sir.
Okay. Just wait, I am presenting a topic. Yes. Is it visible to you?
No, sir. Still no.
Now?
No, sir.
Sorry. It's my fault.
Now it's visible, sir.
Now it's visible? Okay.
Yes, sir. Yes, sir.
Okay, proceed.
Sir, can I start?
Yeah, sure.
Okay. Good evening to all my respected teachers and to all my classmates and my juniors. Welcome to Student Seminar 2021. Myself Goldie Pandey and I am a student of Bangabasi College. I am reading Botany Honors of St. Sixth. Today I am going to represent the PowerPoint presentation on the topic mineral nutrition in plant, which is mentored by our teacher Arpita ma'am. Our respected teacher Arpita ma'am. So now I am going to discuss about it. Sir, introduction part. So, what is plant nutrition? Plant nutrition is the study of the chemical elements and compounds which is necessary for the plant growth and plant metabolism and their external supply. In its absence, the plant unable to complete their life cycles, or that the elements is a part of some essential plant constituent metabolism. The total essential plant nutrition includes 17 different elements. Now moving to the next slide. Techniques which is used in plant nutrition are, there are basically three main techniques which is used in plant nutrition. First one is hydroponic, second one is an alternative system of hydroponic which is known as the nutrient film growth system, and the third one is the aeroponic system. In hydroponic system, the roots are immersed in the nutrient solution and the air is bubbled through the solution. And in the nutrient film growth system, the nutrient solution is pumped as a thin film down a shallow through surrounding the plant's roots. In this system, the composition and pH of the nutrient solution can be controlled automatically. And the nutrient film growth system is an alternative form of hydroponic system and is often used in a commercial production. And the third one is the aeroponic system. Here the roots are suspended over the nutrient solution which is whipped into a mist by a motor-driven rotor. So next slide. Now I am going to discuss about the criteria of essentialities. As proposed by the Erner and Stout 1939 and modified by Erner in 1954, there are three main criteria. The first one is, in the absence of the essential nutrients elements, the plant unable to complete their life cycles or the vegetative or reproductive stage. The deficiency is specific to the element and can be corrected or prevented by supplying that element. The element is involved directly in the nutrition and metabolism of the plant. So next slide. Types. Based on the quantitative requirements by plants, mineral nutrition can be categorized into two types. That one is macronutrients and other is micronutrients. Macronutrients are those elements which are required by the plants in large quantities, that is in excess of 10 mole per kg of dry matter. It includes carbon, hydrogen, oxygen, nitrogen, sulfur, chlorine, potassium. Among these, the carbon, hydrogen, and oxygen are mainly obtained from carbon dioxide and water, and other are absorbed from the soil as mineral nutrition. Micronutrients, these are the nutrients that are needed by the plants in small quantities, that is in less than 10 mole per kg of dry matter. It includes manganese, copper, molybdenum, zinc, boron, chlorine, nickel. So next slide. Sources of essential elements. The plants get carbon, hydrogen, oxygen, nitrogen from the soil, water, and air, and from the fertilizer it gets phosphorus, potassium, calcium, manganese, sulfur, boron, zinc, copper, manganese, iron, chlorine, molybdenum, etc. So next slide. Importance of mineral nutrition. Green plants are autotrophic, that is they can synthesize their organic food from inorganic source. Animals are heterotrophic, that is they cannot synthesize their organic food from the inorganic source and depend directly or indirectly on plants for their food. All plants require basic constraints for synthesis of food and functional functioning of the metabolism. Thank you.
Thank you, Goldie. Good presentation. Now it's time for interaction session.
Yes, sir.
So anyone, Olidi, Devashriti, have you any question for Goldie?
No, I don't have any question.
Okay. Biplabda or Vinati, do you have any question? Sir? Any participant from our students corner, have you any question that you may ask to Goldie or your self-sufficient query? Okay, Goldie, then I want to ask one simple, very simple question.
Yes, sir.
We know that micro elements that are sometimes called the trace elements. Okay.
Micro elements are trace elements.
Yeah. Have you heard about the tracer elements?
Yes, sir. They are found in the traces only. For example, calcium, manganese, and the macro.
What is the function of the tracer element?
Functions of the traces element. No, sir, I have no idea.
Where do you know, man? Where have you got the idea of the tracer element? Where have you got this?
Trace element.
Sir, can I answer the question?
Yeah, sure, Gordon.
Sir, these are isotope elements which are used as molecular markers also, and we have heard the term in photosynthesis where the scientist Robin Hill used carbon isotope to found the role of CO2 in photosynthesis, sir.
Exactly. I think Goldie, you have understood what is the tracer element. So that is the so that is used to indicate or exactly the find out the any pathway. Govardhan, nice mention. Thank you, Govardhan, for your nice answer and for the positive interaction. Thank you. Once again, thanks, Goldie, for your nice presentation. Thank you.
Thank you, sir. Sir, can I turn off the video and audio?
Sure.
Okay, sir.
Our next presenter, Jyotirmoy Das of SEM 4. Jyotirmoy.
Hello, sir.
Hi. Jyotirmoy. Yes, Jyotirmoy. Slides দেখা যাচ্ছে?
Yes, sir. Good evening. Hello everyone and welcome to my presentation. My name is Jyotirmoy Das from Botany Department SEM 4. Today I would like to talk about the nutritional and medicinal value of mushroom. So let's get started. Next slide, sir. Mushroom is the reproductive structure produced by some fungi. Mushrooms have been found effective against cancer, cholesterol present study, cholesterol reduction, isoniemia, asthma, allergies, diabetes. The present study reviews that different aspects of mushroom towards human health benefits such as food, medicines, minerals, drugs, etc. Next slide, sir. My next point is nutritional value of the mushroom. That mushrooms are rich in protein, small amount of carbs, and very small amount of fat. In mushrooms, we have found an unsaturated fatty acid, which is linoleic acid on omega-3 acid. Take a look on this chart. As you can see that the mushroom areas of nutrient like protein, lipid, carbs, etc. However, mushrooms are rich in vitamin B and small amount of vitamin A, D, and also it contains some minerals
potassium, magnesium, sodium, phosphorus, and minorly like zinc, ferrous, etc. Next slide, sir. My next slide name is enzyme inhibiting activity of mushrooms. Tyrosinase is a key enzyme of melanin synthesis, which is found in mushroom, and dopa oxidase activity helps to activate the tyrosinase enzyme for this for this melanin synthesis process. Thus, enzyme inhibiting is an important field of pharmaceutical research, which are useful for treating many diseases. Okay, let's turn now to another point, sir. It's mushroom as a source of dietary fiber. In mushrooms, have a bulk amount of dietary fiber is present, which may help us to control blood lipids, glucose levels, and mainly dietary fiber consumption in our diet can yes, ma'am, help to get
No, please do continue.
sorry, can help to get a good digestive system. Okay, next point, sir. My next slide is antioxidant. As we know that antioxidants are which substances that may protect our cell against from free radicals, which may play a role in heart disease, cancer, cell damage, and other disease. Next point, sir. My next point is medicinal value of mushrooms. That is very important point, that mushrooms can help us to promote our immune function, boost health, lower the risk of cancer, inhibit tumor growth, reduce inflammation, and maintain the oxygenation mechanism of our body. As an example, a diabetic patient easily consume mushroom in its daily diet because of its low caloric and its no starch. So at the end of the topic, as you can, sir, next topic. As you can see that mushroom contains minerals, low caloric. So adding in our daily diets, it helps to maintain our good health. Okay, my presentation is over now. Thank you. Have a nice day, sir.
Thank you, Botany. Have a nice day to you also. So now the interaction session. Is there any question for Jyotirmoy?
Jyotirmoy, you have delivered very nice. Okay. What percentage, what amount of protein is there in mushroom?
Sir, 2 gram.
Percentage means in 100 gram, how much?
Sir, 3 to 4%.
No, not so low amount.
Sir, actually,
it is a higher amount.
Sir,
protein.
নিউট্রাসিউটিক্যাল হিসেবে বলা হয় এত হাই প্রোটিন থাকার জন্য
হাই প্রোটিন সেজন্যই তো ইজ ইউজড অল ওভার দ্য ওয়ার্ল্ড স্পেশাল
নিউট্রাসিউটিক্যাল এজেন্ট
ওস্টার মাশরুমের ক্ষেত্রে দেখেছিলাম স্যার 3 গ্রাম প্রোটিন ছিল, ওই জন্য পার্সেন্টেজ
না এটা আরো বেশি আছে 30% মোর অর লেস 30%. ওকে
পার্সেন্টেজ পার্সেন্টেজ গ্রাম নয়, পার্সেন্টেজ মানে 100 এর মতো। আর
কত গ্রাম
মাশরুমে তুমি নিজেই বলেছিস যে এর ক্যালোরিফিক ভ্যালু খুব বেশি,
হাই পার্সেন্টেজ প্রোটিন, হ্যাঁ বলেছো।
হ্যাঁ
ওকে,
স্যার রাইটলি পয়েন্ট করেছে, ওকে জেনে নাও স্যার যেটা বললেন যেটা কতটা পার্সেন্টেজ থাকে, অনেকটা হাই প্রোটিন থাকে মাশরুমে, সেই কারণেই মাশরুম ডায়েটের চার্টে থাকলে খুব ভালো।
এই প্রসঙ্গে আমি একটুখানি বলি, এটা একটু লাইন থেকে ডেভিয়েট করছি, তবুও বলছি আমাদের মতন গরিব দেশে যেখানে মাছ মাংস দাম বেশি, উই ক্যান রিপ্লেস ইট বাই মাশরুম ইফ দ্য মানে প্রোডাকশন ইজ হাই। প্রোডাকশন অবশ্য ইন্ডিয়াতে অত হাই না, চায়না, তাইওয়ান, জাপান, দে আর ইউজিং ইট, দে আর হাইলি ইউজিং।
আচ্ছা ম্যাডাম, আমার প্রসঙ্গ ধরেই আমি আপনার প্রসঙ্গ ধরেই আমি বলতে চাই, সত্যি কথাই এই চায়না জাপানে এত এত তৈরি করছে, ওরা প্রচুর এক্সপোর্ট করে এবং নিজেরাও প্রচুর খায়। আচ্ছা, আমাদের দেশে আমরা কেন করতে পারছি না?
আমাদের মানে আমরা অন্যদিকে এই দিকটা চিন্তা ভাবনা করে দেখিনি। এটা তো মানে নিউট্রাসিউটিক্যাল, অনেক ডিজিজ কিওর করে। ইটস আ রিয়েলি ভেরি গুড ফুড, এ ক্লাস ফুড।
তাছাড়া আমার মনে হয় লোকেরাও অনেক সচেতন নয়, মানে জানেন না, বুঝেন না।
আমাদের এখানে তো বলা হয় টোড স্টুল, ব্যাঙের ছাতা, এইভাবে এটাকে in China when I went for a workshop I had to face such question why it is called toadstool in India. যাই হোক, এটা অন্য প্রসঙ্গে চলে যাচ্ছি, আমাদের পয়েন্ট।
আচ্ছা
ম্যাম অ্যাকচুয়ালি আমাদের দেশে আবার পয়জনাস হিসাবেও ভাবে, সেই জন্য।
হ্যাঁ ঠিকই টোডস্টুল পয়জনাস হিসেবে। ইউ আর কারেক্ট জ্যোতির্ময়, ইউ আর কারেক্ট।
জ্যোতির্ময়, একটা কথা ছিল আমার, সেটা হচ্ছে তুমি যখন স্লাইড তোমার ভালো হয়েছে, কিন্তু স্লাইড যখন রেডি করবে না, এই যখন পাওয়ার পয়েন্ট করা হয় তখন ফন্ট সাইজটা কিন্তু একটু মাথায় রাখতে হয়। এত ছোট ছোট করে ফন্ট সাইজ দেওয়াটা কিন্তু পাওয়ার পয়েন্টের রুল নয়। আর একটা হচ্ছে ইউ শুড বি ভেরি কেয়ারফুল অ্যাবাউট দ্য স্পেলিংস। তুমি
আমি সেটা বলছি,
স্পেলিংস এত ভুল করেছো, তারপরে সায়েন্টিস্টদের নাম ক্যাপিটালসে দাওনি, এগুলো কিন্তু একটু কেয়ারফুল হবে।
আমি আরেকটা জিনিস বলবো জ্যোতির্ময়, আমি বলে সেকেন্ড স্লাইডে দেখলাম অ্যাগারিকাস বাইস্পোরাস বা কিছু একটা লিখেছিলে, তাই না? হ্যাঁ, সেখানে অ্যাগারিকাসটা ঠিক ছিল, বাইস্পোরাস যেটা লিখেছিলে বিটা কিন্তু ক্যাপিটাল ছিল, এবং তুমি ওটাকে আইটালিক্সে আইদার তোমায় লিখতে হবে, আদারওয়াইজ আন্ডারলাইন করতে হবে। এটা হচ্ছে স্ট্রিক্ট রুল অফ বাইনোমিয়াল নোমেনক্লেচার। এটা পরীক্ষা তোমরা যখন দেবে অবশ্যই এটা কিন্তু ফলো করবে, নাহলে কিন্তু নম্বর ডিডাক্টেড হবে। সো বি কেয়ারফুল
ম্যাম।
ওকে, আদারওয়াইজ ইট ওয়াজ কোয়াইট গুড।
ম্যাম অ্যাকচুয়ালি মোবাইল টাইপ করতে, সেইজন্য হয়ে গেছে।
না, এই মিস্টেকটা কিছুতেই করা যাবে না, মানে সায়েন্টিফিক নেমটা ঠিকঠাক লিখতেই হবে, ঠিক আছে? আদারওয়াইজ ইট ওয়াজ গুড। ওকে,
জ্যোতির্ময় ম্যাডাম যেটা বললেন বা স্যার যেটা বললেন, একদম কিন্তু এটা খেয়াল করবি, যখনই কোনো প্রেজেন্টেশন করতে যাচ্ছি বারবার ক্রস চেক করব যে আমার কোথাও ভুল আছে কিনা লেখায়।
আচ্ছা,
কারণ স্পেলিং মিস্টেক এবং এই ইটালিক্যালি বাইনোমিয়াল নোমেনক্লেচার এইগুলো সে চোখে পড়ে আর কি। হ্যাঁ, সেই কারণেই ম্যাডামরা বললেন। হ্যাঁ, তো এগুলো বলে কারেকশন করার চেষ্টা করি, ঠিক আছে?
হ্যাঁ স্যার,
ঠিক আছে।
ওকে স্যার, ক্যান আই টার্ন মাই অফ অডিও?
Sure. Thank you, Jyotirmoy.
Okay, sir.
So next turn for Madhumati Mondal of Sem 6.
Yes, sir.
Madhumati, can you hear me?
Yes.
Okay, so turn on your video. Yes. Good. মধুমতি,
হ্যাঁ স্যার,
দেখতে পাচ্ছিস তোর প্রেজেন্টেশন?
হ্যাঁ স্যার দেখতে পাচ্ছি।
স্টার্ট করো।
Hello everyone, good evening to all of my respected teacher and my fellow classmates and loving juniors. Myself Botany Mondal, a student of Botany Honours Semester 6, Bangabasi College. Today my topic for this seminar is cancer. Though cancer is a vast topic, I will try to give a little speech on it. So with everyone's permission, I will like to start my presentation. What is cancer? So basically, cancer is abnormal and uncontrolled cell growth among cancer cells that invade destroy the surrounding tissue. Cancer cells are spread via blood and lymph, and this process is called as metastasis. Cancer cells are different from normal cells. Normal cells have a limited lifespan and limited cell division, but in case of cancer cells, their cell division is uncontrolled. Normal cells can contact with other cells, but in case of cancer cells, this property absent. The uncontrolled growth of cancer cells is called tumor or neoplasm. So next, please. Type. There are more than 100 types of cancer, but the most common type of cancer are carcinoma, that is skin cancer, mainly derived from the epithelial cells, and 80% of tumors are carcinomas. Second one is sarcomas, that is mainly bone, cartilage, tendon, and lymphoid cancer. Third one is leukemia. Leukemia is generally known as blood cancer. In leukemias, abnormal increase of white blood corpuscle is occurred. Except them,
Madhumati, I am interrupting. Just say louder. Suddenly sound টা তোর কমে গেল। একটু loud বল বাবা।
okay, sir. Except them, liver cancer, brain cancer, breast cancer, these are also a common type of cancer. Next, please, sir. Causes. Study of cancer cell is called oncology. Oncogenes and tumor suppressor genes are the main responsible for cancer, for causes of cancer. Cancer cause genetic changes also responsible for causing cancer. Chemical and physical agents that cause cancer is known as carcinogen. Except them, heavy alcohol consuming, excess body weight, poor nutrition are also reason for cancer. So next, please. Symptoms. There are many symptoms occur in a person who was attacked by cancer, but most common symptoms are skin color changes, that may be of yellowish or blackish in color, blood present in saliva, blood present in stool and urine also, weak bones, decrease of immunity power, hearing losses are the common symptoms of cancer. So next, please. Treatment. Actually, there is no appropriate treatment for cancer because cancer cells are immortal, but we can prevent this deadly disease by following this process. First one is surgery, that is cut of the tumor. Second one is chemotherapy. This is a process where using of drug are mainly destroy the cancer cells. Radiation therapy, that is high dose of radiation is used to kill the cancer cells. Bone marrow transplant, cryoablation, in cryoablation where extreme cold is used to kill the cancer cells. Please. Here is a graphical representation possibility of cancer before and after. The World Cancer Report said that according to the estimated cancer burden in India in 2018, there are about 1.16 million new cancer cases and more than 7 lakhs cancer deaths. Lung cancer are the most deadliest type of cancer. Pancreatic cancer are the silent killer. Lowest cancer rate in Syria and highest cancer rate in Australia. We celebrate World Cancer Day on 4th February. And next, please. It is one of the most deadliest disease nowadays. Cancer can be very much serious for the people who already attacked by HIV. The goal of all cancer treatment is to win the fight against cancer. The next, please. I am thankful to all of my teacher and my entire department for giving me such a golden opportunity. This is a very interesting topic. I have learned a lot of things about cancer, and these are the references. Thank you.
Yes, মধুমতি না ঠিক আছে, ভালোই বলেছিস। ক্যান্সার হচ্ছে কোন জিনের জন্য তুই তো বলেছিস? অঙ্কোজিন। হ্যাঁ, এই অঙ্কোজিনটা হিউম্যান সেলে কি ফর্মে পাওয়া যায়? অঙ্কোজিন হিসেবে
আমাদের ছোট অঙ্ক হিসেবে থাকে, যা
প্রোটো অঙ্কোজিন, তাই তো?
যা মিউটেশনের কারণে
হ্যাঁ পরিণত হয়। ঠিক আছে, ওকে।
থেরাপিতে না একটু ইমিউনোথেরাপিটা অ্যাড করে দিস, হ্যাঁ মধুমতি, ওটা ভালো, বলা খুব ভালো হয়েছে, তো ইমিউনোথেরাপিটা অ্যাড করে দিস।
ওকে ম্যাম,
ঠিক আছে।
হ্যাঁ মধুমতি, গুড, ভেরি গুড। ওই তোর একটা স্লাইড ছিল না যেখানে টাইপস অফ ক্যান্সার ছিল?
হ্যাঁ স্যার।
ওটা কি 200 টাইপস না 100 টাইপস?
না স্যার, ওখানে অতগুলো টাইপ দেখানো নেই।
না 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 না, তুই লিখেছিস তো 200 টাইপস, মুখে বললি 100 টাইপস, তাই বলছি কোনটা ঠিক?
না স্যার, আমি তো বললাম যে মোর দ্যান 100 টাইপস অফ ক্যান্সার অকার।
আচ্ছা, ওকে ওকে, দেন গুড।
আচ্ছা মধুমতি, আমি একটা প্রশ্ন করব?
ইয়েস ম্যাম।
মানে nowadays we are hearing that cancer is psychosomatic. From stress cancer comes. Is it true? মানে মেন্টাল স্ট্রেস থেকে কি ক্যান্সার আজকাল হচ্ছে? মানে আমরা অনেক জায়গায় পড়ছি এদিক ওদিক যে স্ট্রেস থেকেও ক্যান্সার হতে পারে। সো মানে আমি জানি না, তোমরা নিশ্চয়ই পড়াশোনা করো, সেই জন্যই বলছিলাম।
ম্যাম, ক্যান্সার তো জেনেটিক চেঞ্জের কারণেও হতে পারে। এবার যদি অতিরিক্ত পরিমাণ স্ট্রেস নেওয়া হয় সেক্ষেত্রে আমাদের জিনের
হ্যাঁ, ইউ হ্যাভ কাম টু দি পয়েন্ট, স্পিক আউট ইউর ঠিকই ধরেছে পুষ্পিতা দি, বল।
হ্যাঁ ম্যাম, অত্যধিক প্রেসার ক্রিয়েট হলে জিনের যে কোনো চেঞ্জ তো ঘটতেই পারে, তাহলে
মিউটেশন আসতেই পারে, ক্রোমোজোমাল অ্যাবনর্মালিটিস। আমি এবার একটু হেল্প করছি, ও ধরেছে ঠিকই পুষ্পিতা দি, ক্রোমোজোমাল অ্যাবনর্মালিটিস আসতে পারে। সেক্ষেত্রে যদি ক্রোমোজোমের ওপর জিনগুলো থাকে, জিনের ফাংশনিং ডিস্টার্বড হতে পারে। এগুলো সব মানে এখনো অত ভালোভাবে প্রমাণ পাওয়া যায়নি, কিন্তু আন্ডার মানে গবেষণা লেভেলে আছে যে স্ট্রেস থেকে হতেই পারে মানুষের,
হতে পারে। ওকে,
খুব যে ফুল প্রুফ সেরকম সেইভাবে এখনো সেইভাবে আমরা এভিডেন্স পাইনি। এবার আমি মধুমতিকে, আমার হঠাৎ একটা ছোট্ট প্রশ্ন মনে পড়লো। এত তো ক্যান্সারের থেরাপি, সার্জারি, অমুক তমুক অনেক কিছু বললি, তুই কি জানিস কোন প্লান্ট থেকে ক্যান্সারের ট্রিটমেন্টের গবেষণা চলছে? জানিস কি? যেহেতু বটানি পড়ছিস সেই হিসেবে বলছি আর কি।
ম্যাম, আমি শুনেছিলাম কিন্তু এই মুহূর্তে মনে পড়ছে
না। ইট ইজ এ জিমনোস্পার্ম, শুনে রাখ, ট্যাক্সাস ব্যাকেটা, এর থেকে কাজ চলছে। ওকে ওকে।
হ্যাঁ ম্যাডাম, আমি একটু যোগ করি, ওখান থেকে ট্যাক্সল বলে একটা, হ্যাঁ ট্যাক্সল বলে, হ্যাঁ পাওয়া যায়, সেটাই ক্যান্সার ট্রিটমেন্টে কাজে লাগে।
মধুমতি, তুমি এক কথায় সবাইকে উত্তর দিয়ে দাও, ক্যান্সার হ্যাজ নো অ্যান্সার।
ভালো বলেছে,
একদমই তাই।
এক কথায় উত্তর দিয়ে দাও, ক্যান্সার হ্যাজ নো অ্যান্সার।
স্যার রাইটলি পয়েন্টেড আউট। থ্যাংক ইউ মধুমতি, খুব ভালো প্রেজেন্টেশন হয়েছে, ভেরি গুড ইন্টারেক্টিভ সেশন। ওকে, থ্যাংক ইউ সো মাচ।
স্যার আমি মিউট করতে পারি?
হ্যাঁ হ্যাঁ, অবশ্যই অবশ্যই। আমাদের নেক্সট পার্টিসিপেন্ট মধুমিতা বণিক of SEM 2. মধুমিতা।
Yes sir, am I audible?
Yeah, clearly audible. Thank you for the presentation. Is it visible to you, Madhumita?
Yes sir, yes sir.
So start.
Good evening to all my respected teachers, principal ma'am and all my dear friends. Today I, Madhumita Banik, semester 2 Botany Honours, is going to present a PowerPoint presentation on the topic Cyanobacteria. So second select. Cyanobacteria. Cyanobacteria, also known as Cyanophyta, are a phylum of gram-negative bacteria. They are also called blue-green algae because they are photosynthetic and aquatic in nature. Occurrence of Cyanobacteria. Members of Cyanobacteria are found mainly in freshwater like Oscillatoria, marine like Dermocarpa, and terrestrial like Nostoc. Some are also found in symbiotic associations with different fungi and lichen like Nostoc and Cyctonema. These are the salient features of Cyanobacteria. The vegetative body may be unicellular, multicellular, filamentous or non-filamentous colonial types. In addition to photosynthetic pigment, chlorophyll A, and carotenoids, blue pigment known as C-phycocyanin and sometimes a red pigment known as C-phycoerythrin, that is bilirubins of varying proportions, are present. The reserve foods are cyanophycean starch and cyanophycean granules. Reproduction takes place by vegetative and asexual methods. Vegetative reproduction takes place by cell division, fragmentation, etc. Asexual reproduction takes place by endospores, exospores, akinets, nanospores, etc. Sexual reproduction is absent. So next slide. This shows the cyanobacterian cell. These are the features of the cell structure. So next. Now we come to the chromatic adaptation. As the name suggests, it is the capability of cyanophytes to vary their pigment ratio, especially phycobilins like phycocyanin to phycoerythrin. So next slide. Gaiduka phenomenon. Generally, true vacuoles are absent in the protoplast. A number of vacuoles called pseudovacuoles are distributed throughout the protoplast. When the same species are grown in different wavelength of light shows significant variations in pigment composition. This is called Gaiduka phenomenon. So next slide. Cyanotoxins. Cyanobacterial blooms, generally known as algal blooms, release toxins called cyanotoxins which are very harmful to marine environments as well as humans. When in contact with human beings, it can cause ingestion, dermatitis, rhinitis as well as conjunctivitis. They can also be fatal in extreme cases. Now we come to the applications of cyanobacteria in biotechnology. First, I would like to say something about the role of cyanobacteria in biotechnology. Cyanobacteria have gained a lot of attention in recent years because of their potential applications in biotechnology. More than 14 cyanobacterial genera, including Anabina, Calothrix, Oscillatoria, Nostoc, Microcystis Glyobacter, Aphanocapsa, and Microcoleus, are known for their ability to produce hydrogen gas under various culture conditions. Cyanobacteria have been identified as a rich source of biologically active compounds with antiviral, antibacterial, antifungal, and anti-cancer activities. Cyanobacterial hydrogen has been considered as a very promising source of alternative energy and has now been made commercially available. Cyanobacteria as biofertilizers. Heterocystis cyanobacteria and several non-heterocystis cyanobacteria are known for their ability to fix atmospheric nitrogen. For example, azolla was used as an organic fertilizer in rice cultivation in many countries. Recently, nitrogen-fixing cyanobacteria have been reported to dominate desert crusts worldwide. This is believed to contribute significantly to the fertility of desert soil and may eventually facilitate vegetation of deserts. In addition to these applications, cyanobacteria are also used in aquaculture, wastewater treatment, food, fertilizers, and production of secondary metabolites, including vitamins, toxins, enzymes, and pharmaceuticals. Future research should focus on isolating new cyanobacterial strains, producing high-value products, and genetically modifying existing strains to ensure maximum production of the desired products. So next slide. Lastly, I would like to express my profound thanks and gratitude to Dr. Himadri Bhattacharya Chakraborty, Principal of Bangabasi College, for giving me such an opportunity to do this seminar. I would also like to express my thanks to Dr. Arpita Swarnakar, Head of the Department, the mentor of my seminar work, Dr. Oli Khan Sen, the convener of this seminar, Dr. Argha Sarkar, and all other departmental teachers for guiding me through this seminar. Last but not the least, I would like to thank my family for constantly being by my side, giving unfathomable encouragement, and my friends for cooperating during this seminar. Thank you.
Excellent.
Excellent. Excellent.
This is called
খুব ভালো
fresh and focused presentation.
Yes, yes.
Thank you so much.
Objective was very clear.
Excellent. সুপার যাকে বলে আর ও একদমই সেকেন্ড ইয়ার সবই মানে ফার্স্ট ইয়ার টু
মধুমিতা খুব স্মার্টলি বলেছে, দারুণ।
I have a can I ask questions or go?
Yes, surely.
One small question. You said this cyanobacteria is used as a biofertilizer, so that is a positive point for cyanobacteria দিক থেকে economically. কিন্তু কেমিক্যাল ফার্টিলাইজার না ইউজ করে আমরা কেন এই বায়ো ফার্টিলাইজার ইউজ করছি?
ম্যাম কেমিক্যাল ফার্টিলাইজার্স আমার ধারণায় কেমিক্যাল ফার্টিলাইজার্স ইউজ করলে কোনো ইফ দে প্রডিউস এনিথিং toxic or something, they might affect. Yes ma'am.
You have come to the point. So we are replacing it with the biofertilizers. যদিও এখনো সেইভাবে প্রচলনটা হয়নি, কিন্তু কেমিক্যালকে যদি বায়োফার্টিলাইজার দিয়ে রিপ্লেসমেন্ট করা যায়, আমার মনে হয় সেটা অনেক ক্রপের অনেক ক্ষতি কম করবে। যেমন সাইনোব্যাকটেরিয়াটা তো রাইস ফিল্ডে ইউজ করাই হয়।
Yes ma'am.
রাইস ম্যাচিওর করা। ওকে, থ্যাংক ইউ, থ্যাংক ইউ, থ্যাংক ইউ। এক্সেলেন্ট প্রেজেন্টেশন, এক্সেলেন্ট।
থ্যাংক ইউ ম্যাম।
মধুমিতা।
Yes স্যার।
আমি as per my knowledge is concerned, গাইডিকো ফেনোমেননটা বোধহয় লাইট রিলেটেড। তুমি যেটা উল্লেখ করনি, উল্লেখ করনি কিন্তু হ্যাঁ, লাইট রিলেটেড একটা ফেনোমেনন।
Sir, I have included that in chromatic adaptation, and so I could not include in Gaiduko.
You should have mentioned the light relation. Okay. Anyway, but your presentation is very good. Thank you.
Thank you, sir.
The sir has pointed out rightly. You have used two slides, one for chromatic adaptation and another for Gaiduko phenomenon. So you have to correlate the chromatic adaptation with Gaiduko phenomenon when you will go through the exactly the wavelength of light and its variation of wavelength of light. Only then, in different layers of water, we can say that due to the surface or the bottom layer, whatever the cyanobacterial position is there, but due to the variation of the wavelength, the coloration is different, and that's why the Gaiduko phenomenon is also established in case of cyanobacteria.
Yes, sir. Okay,
Can you name one non-heterocystous nitrogen-fixing cyanobacteria? Can you give an example?
Ma'am, I can guess.
You have mentioned that they may be heterocystous and non-heterocystous. Just don't know. That's why I'm asking you.
Ma'am, I can guess. I don't know the right answer.
Okay, you answer.
No, Anabina has it.
No. It is oscillatoria. It is oscillatoria.
Oscillatoria.
Okay, thank you, ma'am.
Okay, thank you.
Madhumita, I would just like to add one thing, since you are preparing now. So it is a protocol that if you are using any diagram, it should be always labeled. So I found a slide of cyanobacteria which was not labeled. Maybe you have taken it from the internet. Anyway, yes, that is something else. But since you are appearing for examination, be sure that if you are not labeling any diagram, then no marks will be given for that. Anyway, now you are learning, so keep this in mind. Okay.
Okay, ma'am.
This presentation was very good.
Thank you, ma'am.
Keep it up. Keep it up.
And one more point, Madhumita, those two slides. First, one slide was the picture of the image of the cell, and the next slide was the description. These should be merged. Okay, side by side, having along the salient features, you have to reproduce the image also to the visitors or audience. Okay, so that the audience can correlate exactly what is the body structure and what the cell structures. Okay.
Okay,
so
sir.
Always put the images with the write-up in one slide. That is very much easier for the audience to identify and understand the whole situation.
Okay, sir. I'll keep that in mind.
Right. Sure. Very good. Very good. Keep it on.
Thank you, sir. Sir, will I turn off the audio and video?
Sure. So now it's time for another member of same two, Moli Sinha.
Yes, sir.
So Moli, are you there? Yes, it's Moli.
Yes, sir. Good evening to all the respected teachers, principal ma'am, and my dear friends and seniors. Today I, Moli Sinha of semester 2, going to represent my PowerPoint presentation on the topic of cyanobacteria. So let's begin. Cyanobacteria are commonly known as blue-green algae, and they are photosynthetic autotrophs. They contain chlorophyll A, which is similar to green plants and also contain phycocyanin and phycoerythrin. They live in the aquatic environment, including the oceans, ponds, lakes, tidal flats, and moist soils. They exist mostly in colonies and filaments and sometimes as single cells. Next slide, please. Some of the filamentous forms of cyanobacteria can move. For example, the filamentous forms such as Oscillatoria species and Nostoc species rotate in a screw-like manner. They also produce gelatinous capsules which are often lighter than water and therefore helps to keep the algae up near the surface of the water. They reproduce by fission and they are prokaryotic cells. They also lack chlorophyll B and the photosynthetic products are stored in their own form of starch, which is similar to animal glycogen. Next slide, please. Forms of cyanobacteria. So, there are mainly three forms of cyanobacteria. The first one is unicellular or aggregate, example Glyocapsa species. The second one is colonial, example the Microcystis species. And the third one is filamentous form. This filamentous form can be further subdivided into two forms. The first one is the branched and the second one is unbranched. Next slide, please. The cell structure of cyanobacteria. The cell structure of cyanobacteria are very primitive and they are prokaryotic cells with the cell envelope typically for the gram-negative bacteria. The cell wall is composed of two layers. The inner layer, which is the thin and firm, composed of peptidoglycan, and the outer layer of wall is thicker and gelatinous, known as the mucilaginous sheath. And mainly, they constitute the pectic compound. Next slide, please. This is the cyanobacterial cell structure where we can find that cell wall, cytoplasm, DNA, photosynthetic membrane, plasma membrane, and mucilaginous sheath. Next slide. Nostoc. Nostoc is a type of cyanobacteria which grows in water and on damp soil. They are unbranched filaments with barrel-like cells. Certain enlarged cells appear at intervals, which are known as heterocysts. These heterocysts also fix the atmospheric nitrogen. It is transparent and have thick walls. The whole filament is surrounded with a gelatinous material. Each of the two heterocysts delimits in between a number of cells called the hormogonia. Next slide, please. The types of reproduction. There are generally two types of reproductions in cyanobacterial cells. The first one is vegetative reproduction, that is by fragmentation, and the second one is asexual reproduction. The first one is by fission and the second one is by akinesis. Sexual reproduction in cyanobacteria are generally not found. Next slide, please. The importance of cyanobacteria. The importance of cyanobacteria are they can fix the atmospheric nitrogen and also are used as biofertilization. They are used as food in Japan, Chad, and China. They are one of the early colonizers of bare and barren areas. They provide suitable conditions for the growth of other organisms in the hostile environment. Antibiotics can be manufactured from the extract of Limpia, a genus of cyanobacteria. Next slide. Lastly, I would like to express my special thanks and gratitude to my mentor, Dr. Oli Khan Sen ma'am, and also Dr. Argha Sarkar sir for the able guidance and support in completing my presentation. I would also like to extend my gratitude to the Honorable Principal, Dr. Himadri Chakraborty Bhattacharya ma'am, and also the HOD of Botany Department, Dr. Arpita Swarnakar ma'am, for providing me with all the facilities that are required. That's it for my presentation. Thank you.
Excellent. Very good.
Excellent presentation.
Good presentation. Very good presentation. I'm asking a small question. কোনটা বললি যেন খাবার হিসেবে মানে ফুড হিসেবে ব্যবহার হয় একটা এক্সাম্পল দিয়েছিলিস
এক্সাম্পল ছিল না মানে ইয়ে ছিল মানে সায়ানোব্যাকটেরিয়া এগুলোই মানে ফুড হিসেবে খাওয়া হয় ইন সাম কান্ট্রিজ
ঠিক আছে ফুড হিসেবে খাওয়া হয় ইউ আর কারেক্ট কেন রে ফুড হিসেবে কেন খাওয়া হয় কেন কারণটা কি হোয়াট ইজ দ্য রিজন
নিউট্রিয়েন্টস মেবি পেতে পারে
সেই নিউট্রিয়েন্ট গুলো কি কি সেগুলো জানতে চাইছি
আচ্ছা আমি সেরকম ভাবে বলতে পারবো না
ঠিক আছে ভিটামিনস প্রোটিনস হাই কোয়ান্টিটিতে আছে এবং অনেক সময় এই সাইনোব্যাকটেরিয়া এগুলো স্যুপ হিসেবে নিয়ে যাওয়া হয় স্পেস ক্রাফট রিসার্চে ওখানে তো ভারী জিনিস নিয়ে যাওয়া যায় না দিস সাইনোব্যাকটেরিয়াল স্যুপ নিয়ে যাওয়া হয় স্পিরুলিনা হচ্ছে ব্লু গ্রিন অ্যালগি সাইনোব্যাকটেরিয়া হাই প্রোটিন হাই ভিটামিন থাকার জন্য এগুলো কনজিউম করা হয় অ্যাজ ফুড আর সেই জন্য স্পেস রিসার্চে তো হেভি জিনিস ক্যারি করতে পারে না ক্লোরাল ওই স্পিরুলিনার স্যুপ নিয়ে যাওয়া হয়
single cell proteins
due to high protein content. Okay, very good presentation, very good.
Thank you, ma'am.
এবার অন্যরা বলুক।
গুড মর্নিং, ভেরি গুড। ওটি একটা জিনিস বলতে পারবি রে বাবা? মেনশন করেছিস হরমোগোনিয়া।
হ্যাঁ স্যার।
নো অ্যাবাউট দ্য ফাংশন অফ হরমোগোনিয়া।
Sir, they are motile filaments and they are motile filaments এইটুকু জানি আর কি করে ওরা আর ওরা প্রোডিউস হয় হচ্ছে ভেজিটেটিভ রিপ্রোডাকশন থেকে বাস আমি এইটুকুই জানি স্যার।
ওএস ম্যাডাম কিছু অ্যাড করবেন?
কি হরমোগোনিয়া ওদের রিজেনারেশনের কাজে লাগে মানে ফ্র্যাগমেন্টেশন হয় সেখান থেকে নতুন আবার নতুন থ্যালাস গ্রো করে আর তুমি কিছু অ্যাড করলে করো আমি আবার অন্য একটা জিনিসে যাব।
না না ঠিক আছে ঠিক আছে একদম
এইটাই আর কি জানি কি একটা ভেবেছিলাম আমি ভুলে গেলাম মাথায় নেই আউট হয়ে গেল। ঠিক আছে ভেরি নাইস নাইস ভেরি গুড প্রেজেন্টেশন এন্ড কিপ ইট আপ। তোমরা কিপ আপ করো যাতে পরবর্তীকালে আরো ইমপ্রুভ করতে পারো আরো উন্নতি হোক তোমাদের এটাই চাই এই আর কি। ওকে
good Molly for the first time it is a very nice presentation.
Thank you.
Okay so keep it on. Very good thank you.
So can I turn off the camera?
Sure
ও একটা ওকে লিভ ইট
না বলুন না দিদি বলুন।
না ও মলি তুমি যে বারবারই বললে এখানে মানে জিলাটিনাস ক্যাপসুল থাকে বা মিউসিলেজ থাকে ফিলামেন্ট বা কলোনির গায়ে মিউসিলেজটা কি কাজে লাগে? হোয়াট ইজ দ্য ফাংশন ডু ইউ নো?
ম্যাম আই ক্যান ট্রাই মেবি প্রোটেক্ট করার কাজে লাগতে পারে আর ফুড ক্যাপ
প্রোটেক্ট করার কাজে লাগে আর এছাড়া ওয়াটার রিটেনিং ক্যাপাসিটি জলটাকে ধরে রাখে which is also helpful during reproduction asexual reproduction বা যাই হোক। ওকে থ্যাংক ইউ।
Okay Molly. Now it's time for Priyanka Khatua same 4. Priyanka. Yes. Here is Priyanka. Priyanka, am I audible? শোনা যাচ্ছে আমাকে?
Yes sir.
দেখতে পাচ্ছিস? Presentation?
ঠিক আছে।
Okay, start কর।
হ্যালো স্যার।
হ্যাঁ শোনা যাচ্ছে
স্যার ইকো হচ্ছে ভীষণ।
শোনা যাচ্ছে বল শুরু কর।
Hello everybody, good evening. My name is Priyanka Khatua, Department of Botany, SEM 4. Today the topic of my presentation is spice yielding plant. Now I am going to talk about it. So let's get started. Spice. Derived from the Latin word spices meaning specific kind and later goods or merchandise. India is known as home of spices and also land of spices. Spice is a seed, fruit, leaf, bark or other plant part. Spice is used for coloring, flavoring and medicine, perfume and also preserve of food. The next slide. First spice is black pepper. Common name is golmoris. Its scientific name is Piper nigrum. Family name is Piperaceae. Its useful part is diet unripe fruits. We are called black pepper is the king of spices. Black pepper is used in flavoring agent, improve brain function, control blood sugar, lower cholesterol, and cancer fighting properties also available. As you can see this picture. Next slide. Next, I move on cumin. Local name is Jeera. Its scientific name is Cuminum ciminum. Family is Apiaceae. Its useful part is dried fruit, Cumin used in important ingredients, curry, carminative, stimulative, medicine, and also promote digestion. The picture clearly shows. Next, I move on. Clove. Common name is lobongo. Its scientific name is
Syzygium aromaticum. Syzygium aromaticum.
Syzygium aromaticum. Its family name Mitraceae. Its useful part is dry buds. Cloves use is dental-related issue, painkiller, reduce stomach ulcer, and use in curry, etc. As you can see this picture. Next spice is cinnamon. Local name is daruchini. Its scientific name is Gelanicum verum. Its family name Lauraceae. Its useful part diet inner bark of stem. Cinnamon used in curries, making chocolate, perfume, germ killer properties, pharmaceutical, and control neurodegenerative disease. Then we look at the picture. Next, move on. Ginger. Common name is Ada. Its scientific name is Gingiber officinale. Its family Gingiberaceae. Its useful part rhizome. Ginger used in food items, also curries, osteoarthritis, dizziness, and medicine, cold and cough. Next, I move on. Indian cassia. Common name is Tejpata. Its scientific name is Cinnamomum tamala. Its family Lauraceae. Its useful part diet leaves. Indian cassia used in curries and also used in rheumatoid arthritis and medicine also. To conclude, to conclude, it's like to say spices are important role in our life. Thank you all for listening my presentation.
Thank you. Good. প্রিয়াঙ্কা, কোনো প্রেজেন্টেশনের আগে কোনো সায়েন্টিফিক নেম যেটা আমাদের সব সময় পার্সন মানে এটা নিয়ে আমাদের চলতে হয়, সায়েন্টিফিক নেমগুলোকে ঠিক করে স্পেল করতে হবে, এটা প্র্যাকটিস করতে হবে বাড়িতে বেশি করে, ঠিক আছে? যে এক্স্যাক্টলি জেনাসটাকে কি স্পেল করতে হয়, স্পিসিসটাকে কি স্পেল করতে হয়, নিজে না পারলে স্যার ম্যাডামদের জিজ্ঞাসা করে নেবে, ঠিক আছে? এবং অবশ্যই ফ্যামিলি নেমটাও ঠিক করে জেনে নেবে।
ওকে স্যার।
ওকে, গুড প্রেজেন্টেশন। এবার ইন্টারঅ্যাকশন সেশন। কেউ যদি কিছু জিজ্ঞাসা করেন প্রিয়াঙ্কাকে। অর্পিতা দি, your topic.
Priyanka, your presentation was very good, but there are so many spices are there also. Turmeric is also an important spice you have not mentioned, and at least you should mention few names. Okay.
Okay, sir. Thank you, sir.
Yeah, good input from you, sir.
প্রিয়াঙ্কা, এই কোভিড সিচুয়েশনে বলছে স্পাইস আমাদের নাকি ইমিউনিটি বাড়াচ্ছে, এটা শুনছি। আই ডু নট নো, ট্যাক্সোনমিস্ট বা ইকোনমিক বটনি যারা করেন তারা ভালো বলতো। এরকম কি কোনো স্পাইস আছে যেগুলো আমাদের ইমিউনিটি বুস্টিং করে? এটা শোনা কথা, আই এম নট শিওর। হ্যাঁ, বলো
ম্যাম। গোলমরিচ, আমার সাথে যেটা হয়েছে আমি একটা বলি। রিসেন্ট আমার খুব জ্বরে ভুগছিলাম আমি, তো আমি গোলমরিচ, আদা আর দারুচিনি এই তিনটেকে মিক্সড করে গরম জলে ফুটিয়ে খেয়েছি। তো এইভাবেই আমাদের এই যে স্পাইস গুলো খুব ইউজফুল।
আমার নিজের সাথেই এটা খুব মানে হয়েছে, তো আমি খুব
তোমার সেটা রোগটা থেকে কিছু রিলিফ পেয়েছো, তাই তো?
হ্যাঁ ম্যাম, ইভেন ম্যাম এখন আমি কাশিতে ভুগছি, তো আমি কাল থেকে একই করছি, তো অনেকটা রিলিফ পেয়েছি।
আচ্ছা, ওকে ওকে, থ্যাংক ইউ।
Okay ma'am, thank you ma'am.
Okay, Priyanka, thank you for your good presentation. Now we will move on the next presenter. It is Rahul Mondol from SEM 2. Rahul, শুনতে পাচ্ছ?
Yes sir.
আচ্ছা। Is it visible to you? দেখতে পাচ্ছ পরিষ্কার?
Yes sir.
বলো, স্টার্ট করো।
Good evening to my old teacher and my classmate and my senior. I presenting the stellar type of and evolution. The stele is defined as central vascular cylinder with or without pith. The concept of stele as fundamental unit of vascular system was put forward von Techen and Darwin at 1886, who proceed the development stellar theory. This stele of stem as connected with that of leaf by a vascular connection known as leaf trace. Next. This stele is in Pteridophyta can be differentiated into two major groups. One, protostele, the most simplified and primitive type of stele. In stele, the vascular bundle is concentric solid mass and the central core of xylem is surrounded by concentric of phloem cylinder and finally surrounded by layer of pericycle. The example xylotam. The protostele exhibit following forms. Haplostele. This is the most primitive type of stele. In this xylem from a solid, smooth central core which is surrounded by continuous concentric layer of phloem. Example, Senachilenda. The next, actinostele. This is modification of the haplostele and somewhat more advanced in having the central xylem core with ravaging ribs and phloem is not concentrate but present in between the radiating ribs of xylem. Example, Senachilella. The next, Pletostele. In the stem of some species of Lycopodium, the solid core of xylem broke a number of plate-like lobes, more or less arranged like a parallel of one another. The phloem alternates the core of xylem plates. Example, Lycopodium volvuli. The specialized form of protostele is termed as pletostele. Mixed protostele. This type mass of xylem and phloem are uniformly distributed. The scatter group of xylem are embedded with the ground of phloem. Like example, Lycopodium sardina. Protostele with mixed pit. In the certain there are parenchyma cells associated with tracheids. Example, Lepidodendron. It may be divided from the transformation of tracheids into parenchyma and may be first step of transformation of pith. Such type of protostele is termed as mixed protocell. The next, Cyanoptostele, modulator of protostele equals siphonostele, is characterized of Philicophyta. This is modification of protostele. During development of siphonostele, the center core of xylem is replaced by parenchymatous cells. On the basis of branch and leaf gap, Zerifi 1910 is divided into two groups. One of cladosiphonic siphonostele, there are no leaf gaps not found, and phycosiphonostyli, there are leaf and branch gaps are present. Next, origin of pith. Two theories have been proposed explain the origin of pith. Interstellar of origin pith. According to the view, pith is origin of a result of transformation of tracheic elements, the central xylem core into parenchyma, and another is external origin of pith. The hypothesis was supported by Jeffrey 1902, 1910, and 1917. According to his view, Protoset was transformed into Siphonostyli due to mineralization of coronal cells into stellar axis, opening such a leaf and branch gaps, probably provide passage of inhibition of parenchyma. So next, next. JDP 1898 classified Siphonostyli into two type on the basis of possession of phloem. Ectophilic Siphonostyli. This pith is surrounded by concentric xylem cylinder, and next of the xylem the concentrate phloem cylinder. Its mean phloem is the outer side of the xylem. So it example Posidia. An amphiphilic Siphonostyli is surrounded by vascular tissue, and the inner phloem cylinder surrounds the central pith, and then next of the concentrate of xylem cylinder, which is immediately surrounded by outer phloem cylinder. It means phloem is present of the both sides of xylem. Example Marsillia rhizome.
রাহুল, তোমার টাইম অলরেডি শেষ হয়ে গেছে, অতএব তুমি আবার শেষ করো তাড়াতাড়ি।
And some is dicholostyli, polycyclic style and ecostyli, and the evolution of some should grow in when the shoot grows accompanying increased size. There are differentiation of stili. First, apprehension of parenchyma scattered of xylem core, and finally central parenchyma module is developed in protostili. Part of protostili called Siphonostyli, then Siphonostyli modified into the Solenostili, and Solenostili and Siphonostyli with more overlapping gaps is known as dichosis Siphonostyli or dichotostili. The final elaboration of stellar organization in pteridophyta consists in the development of a number of separate stili. Such a stili is known as cyclostili, and another modification of Siphonostyli is the ecostili.
রাহুল, সরি টু সে, আমাদের কিন্তু এখানে স্টপ করতে হবে তোমাকে। অলরেডি 6 মিনিটস ওভার, এবং তোমার এখনো কিন্তু আরো কটা স্লাইড বাকি আছে।
হয়ে গেছে
না, আর একদমই শেষ পর্যায়ে মানে চলে এসেছি। তুমি যদি লাস্ট এই স্লাইড সম্বন্ধে কিছু বলতে চাও, এভলিউশন সম্বন্ধে বলতে পারো। স্টিলির এভলিউশন সম্বন্ধে দু চার কথা বলে শেষ করো।
When shoot grows accompany increase in size, there are internally differentiation in stili. The first apparitions of parenchyma scattered in xylem core, and finally a center parenchymatous module or pith is developed in the protostili. Such type of stili is called Siphonostyli. The method by which metallation came about in a protostili and from in a Siphonostyli is debated question of explained by two hypotheses. One extrastellar and another is interstellar origin pith. Sinachilella and
রাহুল প্লিজ স্টপ হিয়ার। আমার ওকে, ঠিক আছে, তোমার ফার্স্ট টাইম, এটা কোনো ব্যাপারই নয়, হতেই পারে। প্রথম কথা হচ্ছে তোমার স্লাইডের সংখ্যা 19, তোমাকে আমি আগেও বলেছিলাম। সেই 19 খানা স্লাইড নিয়ে 5 মিনিটে বলা আমাদের পক্ষেও সম্ভব না, তোমার পক্ষে সম্ভব নয়, কারোর পক্ষে সম্ভব নয়। তাই সেই কারণেই 10টা স্লাইডের কথাই বারবার বলা হয়েছিল, যে 10টা স্লাইডের মধ্যেই সবাই চেষ্টা করুন, কারণ 10টা স্লাইড বলতে 5 মিনিট কভার হয়ে যায়। এরপর থেকে সবসময় ইনস্ট্রাকশন ওয়াইজ কাজ করবে, এটা হচ্ছে 1 নম্বর। 2 নম্বর হলো হচ্ছে তুমি যদি কোনো বিষয় পছন্দ করো, চুজ করো, যেমন তুমি পছন্দ করেছো স্টিলি এবং তার রিভলিউশন, খুব ভালো একটা টপিক। যখন তুমি এটা পছন্দ করেছো তার মানে তুমি এটাকে সাবজেক্টটা তুমি কিছুটা পড়েছো বা কিছু জানো। তুমি যদি যেহেতু তুমি একদম প্রথমে প্রেজেন্টেশন দিচ্ছ, তোমার দরকার নেই তো ইংরেজিতে প্রেজেন্টেশন দিতে হবে, এরকম কোনো বাধ্যবাধকতা নেই তো। তুমি তোমার নিজের বাংলা ভাষায় যদি ভালোভাবে লেকচারটা দিতে পারো এবং সেটা আমাদের বোঝাতে পারো, তাহলে তুমি একটা নাম্বারও কম পাবে। এখানে কিন্তু আমাদের ল্যাঙ্গুয়েজের জন্য কোনো মার্কস নেই, ওয়েদার ইট ইজ বেঙ্গলি অর ইংলিশ অর হিন্দি অর এনি আদার থিংস। অতএব তুমি যদি এই লাইনগুলো মুখস্থ পরপর না বলে যদি পরিষ্কারভাবে নিজের মতো করে একটুখানি গুছিয়ে স্টিলি কি, স্টিলি কয় প্রকার, সেই যে ভাগগুলো রয়েছে সেই ভাগগুলোর সম্বন্ধে একটা করে ডেফিনেশন দিয়ে তার এক্সাম্পল দিতে এবং ইভলিউশনে তাদের সম্বন্ধে বলতে, তাহলে মনে হয় এটা অনেক ভালো হবে। ঠিক আছে? এরপর থেকে এটা মাথায় রাখবে, কোনো প্রেজেন্টেশন করার সময় ইনস্ট্রাকশন ওয়াইজ তৈরি করবে। 10টা স্লাইডের বেশি তুমি কখনোই 5 মিনিটে বলতে পারো না, কারণ টাইমটা কিন্তু অবশ্যই একটা ফ্যাক্টর যে কোনো প্রেজেন্টেশনের জন্য। ঠিক আছে? মাথায় রাখবে, প্রথমবার কোনো ব্যাপার না, আরো ভালো হবে। চেষ্টা করেছো ভালো, আরো ভালো চেষ্টা হোক, শিখে নাও। যেখানে যেখানে তোমার মনে হচ্ছে সমস্যা হচ্ছে সেগুলো স্যার ম্যাডামদের সঙ্গে ডিসকাস করে নেবে ভালোভাবে। ঠিক আছে? আচ্ছা
রাহুল, আমার একটু সাজেশন আছে তোমার প্রতি। সেটা হলো তোমার ওই স্লাইডের ওই ফন্ট সাইজটা এত ছোট, আমাদের খালি চোখে কিন্তু দেখতে পাচ্ছি, মানে কষ্ট হচ্ছে এরকম ব্যাপার। আর দ্বিতীয়ত, তুমি ডিটেইলস ওরকম স্লাইডে ব্যবহার করতে নেই, ডিটেইলস লিখতে নেই, শুধুমাত্র পয়েন্টস গুলো লিখবে, সেই পয়েন্টস তুমি ডিসকাস করবে। আর অতগুলো স্লাইড করেছো, ওই যে স্যার বললেন, সত্যি 5 মিনিটে 19খানা স্লাইড অসম্ভব। হ্যাঁ, তুমি এখানে 8খানা থেকে 10খানা করতে পারতে যদি পয়েন্ট ব্যবহার করতে। আর একটা কথা বলবো, তুমি ইম্পর্টেন্ট পয়েন্ট গুলো হাইলাইট করবে এবং কালার করবে। আর তোমার যে টাইটেলটা করেছো না, টাইটেলের সাইজটা অত্যন্ত ছোট হয়ে গেছে, সেটাও ভালো করে দেখা যাচ্ছে না। টাইটেলটার সাইজ সাধারণত 40-44 হয় ফন্ট। ওকে।
কালারের ব্যাপারটা আমিও নোট করেছি, একটু কন্ট্রাস্ট কালার, যদিও তোমরা প্রথমবার করছো, পরে ভালো করে করো। কন্ট্রাস্ট কালার করলে সেটা চোখেও একটু পড়ে, মানে কন্ট্রাস্ট ইয়ে থাকলে দেখতেও সুবিধা হয়। ওকে ওকে, ফার্স্ট টাইম হিসেবে ঠিক আছে। বাকি স্যাররা যেগুলো বলছেন সেগুলো ফলো করো।
ঠিক আছে রাহুল, মনে রাখবে যেগুলো সবাই স্যার বললেন এগুলোর কাছে মনে রেখে ফার্দার আরো ভালো প্রেজেন্টেশন নিশ্চয়ই হবে। ওকে, থ্যাংক ইউ রাহুল।
নেক্সট
হ্যাঁ,
সালমা।
তুমি তোমার ভিডিও অফ করে দাও। হ্যাঁ, নেক্সট সালমা জাহাঁ, সেম 4। সালমা। হ্যাঁ সালমা, শোনা যাচ্ছে তো সালমা?
I am audible.
Yes, clearly audible. Is it visible to you, Salma?
Yes, sir. Good evening to all of the respected teachers, seniors, juniors, classmates. My name is. Hello, ma'am.
Yeah, Salma, please.
My name is Sandhya, Department of Botany. Today my topic is nutritional and medicinal value of mushrooms. Next slide, sir. First of all, the importance of mushrooms in our lives is immense. There are many people in this world who cannot afford to eat nutritious food. They can easily take mushrooms as food because mushrooms are very nutritious. It is also very readily available. Not only do we get a variety of nutrients from mushrooms, but its medicinal importance is immense. The next slide. Nutritional values of mushrooms. Mushrooms is considered to be a complete health food and suitable for all age groups, child to aged peoples. Mushrooms are rich in proteins, dietary fibers, vitamins and minerals, and antioxidants. This can have health benefits. Mushrooms is also a good source of vitamins. High levels of riboflavins, niacins, proteins, and traces of vitamin C, B1, B12, B, and E. There are also different types of nutrients found in mushrooms. These carbohydrate, energy, calcium, iron, magnesium, potassium, sodium, zinc, copper, etc. Mushroom is considered to be a complete health food and suitable for all age groups. So next slide. Such as some mushrooms, this picture is on the slide: Agaricus biscorus,
Iodotus, Sajarkazu, Valvariella, Agaricus, Tamensis, etc. We get our nutrition from all these types of mushrooms. So next slide. Mushrooms also have medicinal significance. Since thousands of years, edible fungi have been revered for their immense health benefits and extensively used in folk medicine. Specific biochemical compounds in mushrooms are responsible for improving human health in many ways. Sir, next slide. The bioactive compounds include polysaccharides, terpenoids, low molecular weight proteins, glycoproteins, and immunomodulating compounds. Hence, mushrooms have shown to promote immune functions, boost health, lower the risk of cancer, inhibit tumor growth, help balancing blood sugar, cardiovascular protector, ward of viruses, bacteria, and fungi, reduce inflammation, and support the body detoxification mechanism. Many traditional used mushroom from auricularia, ganoderma, tremella, etc. To possess significant medicinal properties. Sir, next. Such as some mushrooms, this picture is shown on the slide. Lucidium, Simensis, auricularia, auriculus, versicolor, etc. From all these types of mushrooms, we get a variety of medicinal properties that play a very important role in our lives. Thank you, sir.
Thank you, Sarma. Presentation, but not a single scientific name is it being in italics. That's the main fault.
Hello, sir.
Sarma, বল।
হ্যাঁ স্যার, বুঝলাম না, আরেকবার কোশ্চেনটা রিপিট করুন।
বাট কোশ্চেন আমি কিছু বললাম তোর স্লাইডে যে তোর প্রত্যেকটা প্রেজেন্টেশনে খেয়াল কর এই যে সায়েন্টিফিক নেমগুলো রয়েছে একটাও ইটালিক্স করা হয়নি কিন্তু দ্যাট ইজ এ মানে বিগ ফল্ট দ্যাট অলরেডি এস ম্যাডাম অলরেডি মেনশনড ইন আ প্রিভিয়াস প্রেজেন্টেশন। দেখতে পাচ্ছিস?
হ্যাঁ স্যার।
হ্যাঁ, এটা মিস আছে। এই মিসগুলো
আচ্ছা অর্ঘ, ওই মোবাইলে কি ইটালিক্স করা যায়? একবার ওরা বলছিল মোবাইলে নাকি ইটালিক্স করা যায় না, তাই কি?
আন্ডারলাইন তো করা যায়।
হ্যাঁ, আন্ডারলাইন তো করা যায়।
আন্ডারলাইন করা যাবে, ঠিক।
আন্ডারলাইন করা যায়।
ঠিক আছে সালমা, গুড। আমি অনেক মাশরুম নিয়ে অনেক কথা হচ্ছে, বিপ্লবদা আমাকে অনেক ভালো জানে মাশরুম সম্বন্ধে। আমি শুধু একটা জাস্ট কথা সবাইকে না বলে পারছি না, মানে এই মাশরুমের নিউট্রিশনাল ভ্যালু, মানে এই নিয়ে অলরেডি অনেক ডিসকাস করলেন সবাই, আমরা অনেক কিছু বলছি। একটা সবাই জেনে রেখো, মানে এই মাশরুম যেরকম সত্যি সত্যি মানে আমাদের স্টেটে বা আমাদের দেশে এখনো অব্দি সেই অবহেলিত খাদ্য চার্টের মধ্যে এখনো নেই আর কি, সেটা ডায়েট চার্টে এটা একটা ফল্ট, ঠিক কথাই। তবে মাশরুম কিন্তু এমপ্লয়মেন্ট জেনারেশনে একটা বিশাল হেল্প করছে এবং কাজ করছে। এবং তোমরা জেনে রাখো একজনের নাম, সেটা তিনি আমাদের সকলেরই পরিচিত, তিনি প্রফেসর কৃষ্ণেন্দু আচার্য, ফর্মার হেড ডিপার্টমেন্ট অফ বটানি, ইউনিভার্সিটি অফ ক্যালকাটা। মানে হি হ্যাজ marvelous job about mushroom and fungi and mushroom cultivation নিয়ে ওনার প্রচুর ভালো ভালো পেপার, জার্নালস, যাদের ইম্প্যাক্ট ফ্যাক্টর আর সো হাই ইন ইন্টারন্যাশনাল লেভেল। এবং উনি বহু এরকম ফার্মের সাথে রিয়েলি ইনভলভ, যেখানে কিন্তু ইভেন অ্যালং উইথ ওয়েস্ট বেঙ্গল বায়োডাইভার্সিটি বোর্ড উনি সেই ফার্মের সঙ্গে যুক্ত হয়ে সেই ফার্মগুলো যাতে এমপ্লয়মেন্ট জেনারেশন করতে পারে সেখানে ছেলে মেয়েদের শিখিয়ে এসছে। এরকম অনেক ছেলে মেয়েরা আছে যারা কোনো কাজ পাচ্ছে না, তার এমপ্লয়মেন্টের জন্যই মাশরুম রেসপন্সিবল। এটা তোমাদের শুনলে মনে হবে যে এটা কোনো এরকম হতে পারে নাকি, বাট যদি তোমরা খোঁজ নাও, যদি এর অ্যাপ্লিকেশন সম্বন্ধে জানো, তাহলে তোমরা বুঝতে পারবে। ইভেন ইউ ক্যান সার্চ অ্যাবাউট দ্য নেম অফ প্রফেসর কৃষ্ণেন্দু আচার্য ইন গুগল, তাহলে তোমরা বুঝতে পারবে হোয়াট টাইপ অফ মাশরুম কাল্টিভেশন ইজ গোয়িং অন only in our state but throughout the India। এবং ওনারা এটাই চেষ্টা করে যাচ্ছেন যে এই কাল্টিভেশনটা যেন মানুষের কাছে খুব ভালোভাবে পৌঁছায়, মানুষকে এমপ্লয়মেন্ট দেবে এবং মানুষকে ডায়েট দেবে, সবকিছুই দেবে। এবং সেই কারণেই আমরা যেই গোল্ডেন ক্রপ বলে আমরা খুব চেতাবিলি করি, সো মাশরুম ইজ ওয়ান অফ দ্য গোল্ডেন ক্রপ, যদিও বলতে পারি না, বাট অলসো এ গোল্ডেন ব্যালেন্স ডায়েট অবভিয়াসলি। হ্যাঁ, তাই মাশরুম কাল্টিভেশন পড়া এবং মাশরুম নিয়ে জানা এটা আমাদের সকলের পক্ষে খুব প্রয়োজন এই মুহূর্তে। So it is a very good topic, and Salma, good presentation. Thank you.
Can I turn off my audio and video?
Sure. Our next presenter, Shamrat Mollik Chaudhary, SEM 2. Shamrat from SEM 2. Shamrat.
Yes, sir. Good evening to all our respected teachers and our principal ma'am, and to my friends and seniors. I am Shamrat Mollik Chaudhary of SEM 2 Botany Honours. Today I am going to present a presentation about mycorrhiza and its application. So next slide. Mycorrhiza is a symbiotic association between a green plant and a fungus. The autotrophic green plant acts as a carbon source for the fungal part, and the fungal part gives it mineral nutrition to the plant. Botany was to first recognize this association, and Frank, a German botanist, named it mycorrhiza. So next slide. Peterson and Sarkar, 1994, classified mycorrhiza into 7 distinct types. First is ectomycorrhiza, second vesicular arbuscular mycorrhiza or VAM, third is ectentomycorrhiza or arbutoid mycorrhiza, fourth ericoid mycorrhiza, fifth genetoid mycorrhiza, sixth orchidoid mycorrhiza, and seventh monotropoid mycorrhiza. So next slide. Ectomycorrhiza. Ectomycorrhiza, commonly called is shooting mycorrhiza, are found in 3% of plants in forests of temperate, especially pine, birch, and oak. It forms harticnate with the intercellular space of root cortex and form a mantle or sheet. So next slide. VAM or vesicular arbuscular mycorrhiza is a type of endomycorrhizal association where both vesicle and arbuscular are developed, where the vesicle part produce intercellularly and stored materials like phosphorus and other minerals, and the arbusculars stored foods as oil drops and mostly polyphosphate. Vesicular arbuscular mycorrhiza act as good biofertilizer. So next slide. Ectentomycorrhiza or arbutoid mycorrhiza. Some member of family Ericaceae are member of the other family Helicales have mycorrhizal which are intermediate between ecto and endomycorrhiza. Though the mycorrhizal type is found to be associated in arbustus, so this mycorrhiza called arbutoid. Ericoid mycorrhiza. Ericoid mycorrhiza is found in different member of Ericaceae like Erica and Rhododendron. They are not obligate symbionts and can be cultured. So next. Genetoid mycorrhiza is a member of Genetiaceae like Genatilla, Blackstonia get colonized with fungal hyphae within 2 weeks of germination and form genetoid mycorrhiza. Orchidoid mycorrhiza mycorrhizal association is obligate for the member of orchid for germination of their seeds. This is obligate for the orchid species. Monotropoid mycorrhiza. Monotropoid mycorrhiza is commonly act as a bridge between pinus and a saprophytic herb that is monotropa and the fungus which is associated to form this mycorrhizal association that is boletus. So next slide. Mycorrhiza is very much essential in agriculture field. The mycorrhizal association helps in formation of dichotomous branching. Ectotopic mycorrhiza helps in uptake of minerals, act as a reservoir. They are in nutrient deficient soil. The mycelial association help in absorption of calcium, phosphorus, sodium, zinc, and etc. Mycorrhizal association reduce soil erosion, improve soil aeration. It improves the water level in soil for the soil. So next slide. Role of mycorrhiza in forestry. Mycorrhiza plays an important role to establish forest in unfavorable location or barren land. The application of mycorrhiza fungi in forest bed enhance the formation of mycorrhizal association that prevent the entry of fungal root pathogen. Mycorrhizal association has been shown the auxin production enhanced during the dichotomous branching. So next slide. We know we use chemical fertilizer in soil, but the chemical fertilizer reduce the fertility part of the soil. So it is helpful that mycorrhiza we used as a biofertilizer. Mycorrhizal fungus increase the yield of crops by 30 to 40% and also produce plant growth and promoting substance. BAM fungi hyphae enhance the uptake of phosphorus and other nutrients that are responsible for plant growth. It also enhances the growth of black pepper. So we all know that fungal association sometimes pathogenic for plant life, but the mycorrhizal association is only the association which is non-pathogenic for the plant. There is a mutual relationship between them. And thank you, sir. I would like to thank I would like to thank our principal madam, Dr. Himaji Chakravarti Bhattacharya, and our HOD Arpita Sharnokar ma'am, and our mentor, Dr. Debashree Gupta, and Dr. Argha Sarkar. And I would like to thank my family and friends. Thank you.
Thank you, Samrat. Good presentation. Now it's time for introduction.
Samrat?
Yes, sir.
Samrat,
Yes,
is mycorrhizal association found in all plants?
Sir, 90% of land plants is showed VAM like vascular avascular mycorrhizal association.
90%. More than 90%, 95%. Is it found in aquatic plants?
No, sir. It's mostly found in land plants or seed plants.
Okay. No, it is not found in aquatic plants. Name one family where it is not found. Terrestrial plants.
In terrestrial land, it not found. Where?
Yes.
Sir, it is not Chenopodiaceae family.
Yes. Okay. Thank you.
Very good.
Thank you, sir.
Well, Samrat, presentation was very good and the interaction session was good. Thank you for your presentation.
Thank you, sir.
Next, it's turn for Sanghamitra Mukherjee of SEM 6. Sanghamitra?
Yes, sir. Good evening to our principal ma'am and all the esteemed teachers and my fellow students and juniors. My name is Sanghamitra Mukherjee, student of SEM 6. Today my topic of presentation is mineral nutrition in plant. So next slide. Mineral nutrition. Why are they needed by plants? Plants can make their own food through photosynthesis, which are in turn consumed by humans and animals. Thus, the mineral nutrients are very important for plants. These chemical elements and components necessary for plant growth, plant metabolism, and their external supply. In its absence, plant is unable to complete a normal life cycle and may be hampered in many ways. Plant nutrition includes 17 different elements. Those can be obtained by plant from various sources. So next slide. From air, carbon, hydrogen, and oxygen are obtained. These are also known as mainly are obtained as dissolved such as NaCl, Na2SO, and from soil. Macronutrients such as nitrogen, potassium, calcium, sulfur, magnesium, and micronutrients such as iron, boron, chlorine, magnesium, zinc, copper, nickel are obtained. Moving on to the next slide. These minerals are absorbed by plants through two ways, through roots from soil by cation exchange, where root help pump hydrogen ions into soil through proton pumps. The structure and architecture of the root can alter the rate of nutrition uptake. And through leaves from air, they uptake by stomata by means of respiration. The next slide. Functions of different minerals. Nitrogen, found in chlorophyll, ammonia, amino acids, protein, nucleic acid, and organic acid. Phosphorus has a role in nucleic acid, the building blocks of genetic code material in plant cells. Potassium has a role in guard cells of stomata. Magnesium, certain enzymatic reaction requires magnesium as cofactors. It also plays a vital role in plant cells as it appears in the center of the protein molecules. Sulfur, found in sulfur-containing amino acids such as methionine. Calcium are required for calcium peptide, essential in cell wall development and also as a cofactor of certain enzymatic reaction. Next slide. Uh, boron. Boron developed of younger plants parts of boron helps in development of younger parts of plants such as root tips and plays an important part in flowering stages of plant. Iron is used in biochemical reactions that form chlorophylls and are responsible for reduction of nitrogen from nitrate to aminical nitrogen. Manganese is an enzyme in enzymatic reactions. It involves ATP, also activates several enzymes, and is involved in processes of electron transport system photosynthesis. Copper is a component of many enzymes and part of protein in the electron transport system of photosynthesis. Zinc involves activation of several enzymes required for synthesis of indolic acid, a plant growth regulator. Molybdenum is constituent of two enzymes involved in nitrogen metabolism, and chlorine plays a possible role in photosynthesis and may function as a counter ion in potassium flux that involves cell turgor. Deficiency in the next slide. Element present at lower level or concentration cause deficiency. Deficiency of one element may be the symptom of toxicity of another element. Effects may vary from subtle depression of growth rate to obvious stunning, deformities, discoloration, distress, and even death. Visual symptoms of distinctive identification are rare, and they are multiple and moderate. Next slide. The deficiency of certain elements such as nitrogen results in stunted growth, slow growth, and fluorescence.
Shomamitra, time is already up. You just conclude. Okay.
Thus, yes, sir. Phosphorus, potassium, boron, zinc all can cause several different types of effects due to deficiency. Thus, nutrition in plants are very important and could be added to prevent these things. That's it. I would like to thank our principal man, our HOD, Dr. Urpita Sharnukar ma'am, and all our seminar and for letting.
Thank you, Shomamitra. Thanks for
Thank you.
Very good. Some sound problem is being happening, but it's not your point. Don't worry for that. It's good. I have one legitimate question regarding this slide. Okay. You have given a picture. Sorry, you have given a picture here. এখানে যে ছবিটা দিয়েছি দেখতে পাচ্ছিস তো? Okay. Here it is mentioned production of ROS under salt stress. Here it is mentioned effect of salt stress, ionic stress, osmotic stress. Several terms are related to stress. Stress biology is another branch of physiology where stress physiology we have. Do you know what is stress? And how is it related to the mineral nutrients?
I'm sorry, sir. I think I confused it with another similar looking photo. The one I wanted to download was not this one, I
But
think.
that font should not be highlighted. ওকে, কখনো এরকম কোনো ছবি দিয়ে ফেলবি না যে ছবি আমি নিজেই জানি না, হ্যাঁ ব্যাখ্যা করতে পারবো না। খুব স্বাভাবিক, তোর পক্ষে এখন এই অসমোটিক স্ট্রেস, সল্ট স্ট্রেস এগুলো এক্সপ্লেন করা পসিবল নয়। তাই এখানে একটা যে ছবিটা দিলে এখানে ঠিক হতো সেই ছবিটার বদলে এখানে একটা ভুল ছবি এসে পড়েছে। ঠিক আছে, এটা খেয়াল রাখবি, এগুলো কিন্তু খুব মানে একটা মানে মারাত্মক ভুল হয়ে যায় এগুলো। হ্যাঁ ঠিক আছে, স্টিল এমনি স্লাইডস গুলো দেখতে ভালো হয়েছে, প্রেজেন্টেশন ওয়াজ গুড, আরো ভালো হবে। থ্যাংক ইউ। নাও ইন্টারঅ্যাকশন সেশন, যদি প্রশ্ন করেন সম্ভবত। So no any question? Okay, thank you Sanghamitra. Thank you so much.
Sir. So I'm turning off my camera.
Sure. Now it's time for Shayok Pal from SEM 2. Shayok.
Sir, am I audible?
Yes, clearly audible.
Okay. Good evening, all of you, respected Principal Madam, and all my beloved professors of Department of Botany, Bangabasi College. I am Shayok Pal of Botany Honors Semester 2, and shall be presenting a presentation on progymnosperms and its phylogenetic importance. Next. Okay. Now, why are we doing this presentation? Now, we see big trees and all kinds of trees around us, but we don't wonder how they have come into existence. Now, this is the main purpose of this particular presentation, to see how these big trees have evolved from a certain point of time when the earth was still under the process of formation. Now, what are progymnosperms? Progymnosperms are an extinct group of woody spore-bearing trees or shrubs that is believed to have evolved from pteridophytic members such as the trimerophytes, and eventually gave rise to progymnosperm over the course of time. They possess gymnospermous anatomy but reproduce by pteridophytic methods, and to some extent they also provide the precursor to the reproduction of seed plants like gymnosperms. Okay, let us go and have a look at the discovery of progymnosperms. It began in 1911 with the discovery of scalizylon wood by Russian paleontologist Mikhail Dmitrievich Zalsky from Donets Basin. It looked very similar to conifer wood. It was also noted that the ferns of genus Archaeopteris were often found associated with the fossils of scalizylon. It was Charles B. Beck in the 1960s that he discovered the Archaeopteris leaves from the upper Devonian beds of New York and proposed that the wood scalizylon and Archaeopteris belongs to the same plant, now collectively known as Archaeopteris itself. Next slide. This discovery led to the establishment of a new class of vascular plants called Progymnospermopsida that possessed gymnospermous anatomy but reproduced by free sporing pteridophytic methods. Now let us look at the class Progymnospermopsida. It has got three orders: Aniriophytals, Archaeopteridals, and Protopetals. The order Protopetals is still not a very prominent ones, but the Aniriophytals and the Archaeopteridals are the main ones. The following are the general characteristics. I will not discuss them right now. I'm coming to it a little later. Next slide. Okay. Now, let us look at the phylogeny of progymnosperms. Any plant species or any animal species has got a certain point of its origin. Things just don't come by itself. They should have something in the past from which they evolve. Like that, progymnosperm has got the common pteridophytic ancestor. Let us start from class Rhiniophyta, which is a group of extinct pteridophytes. They are earliest vascular land plants, and they lacked leaves. They were sporophytic and had photosynthetic branches in the form of telome truss. They were dichotomously branched, and they were protostillate. Next slide. Now, the next class we will be discussing is a Trimerophytopsida. This is another pteridophytic class, and its members are superficially similar to rhinophytes. They evolved around the time almost similar to that of rhinia, just after it actually. They had pseudomonopodial branching, and they had dichotomous lateral branches, um, and they possessed almost similar anatomy to that of the rhinophytes. Now, these trimerophytes are of great evolutionary importance as the fossils that are close to ancestry of ferns and spenopsids, and also progymnosperms. Now, we will go into the class Progymnospermopsida. Now, the reason why we have visited the Trimerophytopsida and coming to Progymnospermopsida is that even though we have much well developed pteridophytic members that came after the Trimerophytes, but this particular order under Progymnospermopsida, the order Anerophytals, they were very similar to this Trimerophyte. It is believed to have evolved from the Trimerophytes. They were protostele with a vascular system, and this protostele was commonly lobed. They and the main thing about that, they underwent some kind of secondary growth in their vascular bundle. For instance, they added wood to their stem and become shrubby looking vines. They were not trees, but they were vines. They had mesarc or or centrarch primary xylem. They were dichotomously or pseudomonopodially branching. They also had frond-like branches and bore fusiform sporangia clustered all over their specialized branches or leaves. Next slide. Okay, now we will go into the order Archaeopterygials and why they are considered to have ultimately given rise to the gymnosperms. They are eustellate with a ring of mesarc primary bundle surrounding their pit. They had pycnoxylic wood. Their secondary tracheids have circular bordered pits. Now, these circular bordered pits are not exactly like that of modern conifers or gymnosperms. They were almost similar but a little different. Fertile leaf is dichotomously branched, telomeres bearing rows of sporangia on a axial surface.
Time is up, so just conclude the matter.
Okay. I would like to go to the next slide. It was Beck in 1971 that he proposed the following 3 points. Both conifers and Archaeopteryx possess pycnoxylic vascular bundle and recurrent leaves on branches. The determinate axis of Archaeopteryx may have given rise to long and dwarf shoots of conifers. The simple leaves of coniferophytes and fronds of cycadophytes evolved from primitive preforms of Anerophytals, which is another pteridophytic characteristic. The next slide, sir. Now, Caloner and Petit made the following observations in 1987, which proved that even the reproductive pattern was slowly developing from that of the pteridophytes to progymnosperm and finally into gymnosperms. There is a probability of megaspore number being reduced to a single tetrad. The following are the observations. Botsrodendrostrobus, this is a lycopsid with 4 equalized megaspores. Bensonites, these have 2 larger and 2 smaller megaspores. Archaeosperma, which is another archaeopteridal, has 2 larger and has 1 large conspicuous and 3 small megaspores. I mean non-functional megaspores, just like what we found in modern conifers. Okay, it is a very interesting discipline of plant sciences, namely phylogeny. When it comes to progymnosperm, the level of curiosity increases by several fold, and we start to think exactly how the bygone Earth looked like with its then existing biodiversity, climatic, and atmospheric conditions. Progymnosperms from pteridophytes, preceding anatomical and morphological changes, and established as prominent members of prominent constituents of early forests during the Middle Devonian era and gave rise to gymnosperm eventually. So that's how we get to know how trees and eventually the angiosperms might have come into existence. And following is the bibliography. And I would like to thank all of you.
Thank you, Shahrukh. Your content is very good and your understanding is very good, but please delimit your time. Time constraint is very much an important factor in case of PowerPoint presentation. Now you have already 1.5 minutes passed from the scheduled time. So you have to conclude the total matter, whatever be the content. You have to conclude the matter in such a way so that the 5 minutes max be a maximum time for your presentation. Okay, so keep it in mind for the later time. But up to this, it was a good presentation and the content was good. Now it is come to the interaction session. Have you any question from Shayok or Shayok from anyone? Devashriji?
Shayok, in most of the plants of progymnosperms and also in rhinopsida or gymnosperms, what types of vascular bundles are found?
Sir, in most of the progymnosperms, you get kind of vascular bundle, but only in case of aniriophytals, they were protostele, but they were little different from that of primerophytes because they underwent the secondary growth, kind of that, not exactly that, but kind of that, and they became like shrubby trees.
Okay. Thank you.
Shayok, good presentation.
Thank you, ma'am.
And what's the type of wood found in progymnosperms?
Say, ma'am, it is a pycnoxylic wood. That means it is very compactly.
Yes.
It is internally very compact, and it had. I mean, the tracheids of this particular class had bordered pits that were also there in modern conifers and cycads, but these are not exactly the same. They were like a precursor to that. Even though there is a much debate on whether it has evolved from the progymnosperm only or it is a polyphyletic in origin, but anyway, it can be said from the observations which were done by Petit and also Charles Beck.
Okay, thank you. Good.
Understanding of the subject. Okay, good interaction session. Thank you, Shayok.
Thank you, sir. Thank you very much. Have a nice day. Can I put my video off and sound off now? Okay.
Now it's time for Sharon Shatra from Sem 6. Yes, Sharon, am I audible? শোনা যাচ্ছে আমাকে? Sharon?
Sorry, sir. My phone on the Yes, sir.
Sharon, start.
Good evening, everyone. I am Sharon. Good evening, everyone. I am Shayon. Now I am going to give a PowerPoint presentation on crassulacean acid metabolism. So next. Crassulacean acid metabolism, in short CAM, is a pathway for carbon fixation mainly observed in the xerophytic plant. The term crassulacean, it comes from the family Crassulaceae. Apart from family Crassulaceae, it is also have been found in plants of several family like Cactaceae, Euphorbiaceae, etc. So next. As the xerophytic plants, they live in a very dry environment. The water supply to the plant body is very insufficient, and due to the high temperature, the evaporation rate is also high. So because of both reasons, this plant often suffer from dehydration. But in CAM process, the plants, they open their stomata at night, absorb the carbon dioxide and convert the carbon dioxide into the malate and store it for the next day. In the next day, they utilize this malate into photosynthesis without opening the stomata. So the photosynthesis occurs regularly without losing any significant amount of water. So next. So next slide. The whole CAM mechanism has two parts. The first one is dark acidification, and the second one is light deacidification. The dark acidification process it occurs at night, and it has three steps. At the first step, the carbon dioxide it enters into the mesophyll tissue of the lips through the stomata, and with the help of water and carbonic anhydrase, it is converted into carbonic acid H2CO3. But the H2CO3 is split into hydrogen and bicarbonate ion H plus and HCO3 minus ion. In the second step, the bicarbonate ion it reacts with the phosphoryl pyruvate PAPE to produce oxaloacetic acid, and this reaction is catalyzed by PAPE carboxylase enzyme, and one molecule of inorganic phosphate is also released. The source of the PAPE is a storage glucan that is produced in the Kelvin cycle. The storage glucan is first converted into glucose 6-phosphate, and the glucose 6-phosphate is ultimately converted into PAPE. In the third step, the oxaloacetic acid is oxidized into malate by NADPase with the help of NADPase malate dehydrogenase enzyme. The malate is then stored into the cell vacuole as malic acid. So next slide. The next part is light deacidification, which occurs at day, and it also has three steps. In the first step, the malic acid it enters into the cytoplasm from the vacuole, and it is ultimately converted into malate. The malate is then converted into pyruvic acid or pyruvate and carbon dioxide with the help of malate decarboxylase enzyme. The carbon dioxide directly enters into the Kelvin cycle to participate in carbon assimilation. But the pyruvate or pyruvic acid, it is converted into phosphoryl pyruvate with the help of pyruvate orthophosphate dikinase enzyme. And the PAPE is then converted into 3-phosphoglyceric acid via reverse glycolysis. This 3-PGA ultimately enters into the Kelvin cycle. After this, the Kelvin cycle proceeds as usual. So next. This is a schematic representation of CAM pathway. We can see that at night, the carbon dioxide enters into the leaf, and it is ultimately produce malate. And in the morning, the malate is utilized in the Kelvin cycle, and the starch produced from the Kelvin cycle ultimately is converted into PAPE, which is then again used in the dark acidification. So next, ecological significance. The CAM process is highly beneficial for the xerophytic plant, because the xerophytic plant they live in a very water stress conditions. But due to this process, they keep their stomata closed, so the evaporative water loss is reduced, and because of this reason, they have a upper hand in survival in the arid environment like deserts, drought condition, etc. This was the main significance of the কিছু বলছেন? This was the main significance of the CAM. It also have some minor benefits, like some cactus they can maintain their dry weight, and they can efficiently use the incident light. So next, I like to thank our principal ma'am, professors of my department, my classmates, my family, and my mentor, Dr. Argha Sarkar, for giving me this golden opportunity to present myself and helping me to complete the presentation. So next. Here is some book and some website that I have consulted to make this presentation. That's all. Thank you. And if anyone have some inquiries, please carry on.
Good, Sharon. Thank you for presentation. And now
ভালো হয়েছে প্রেজেন্টেশন।
থ্যাংক ইউ হয়েছে বলে যদি কেউ প্রশ্ন করেন, দিদি কিছু প্রশ্ন শারনের জন্য?
না, প্রশ্ন কিছু নেই, ঠিকঠাকই তো বলেছে, মানে যা যা বলার বলেছে সব।
থ্যাংক ইউ ম্যাম।
বুঝে বলেছিস, এটা ভালো। বুঝে বলাটাই সব থেকে দরকার প্রেজেন্টেশন। ঠিক আছে শারন।
So,
Thank
sir,
you so much.
can I turn off the audio and camera?
Sure. Next, Shubhodip Bhuya from SEM 4. Shubhodip.
Hello, sir.
Hi.
Good evening, everyone.
দেখতে পাচ্ছিস শুভদীপ?
হ্যাঁ, sir.
শুরু কর।
So, I'm Shubhodip Bhuya from Botany Honors 6 SEM 4. Today I will discuss about ecological succession. Ecological succession, what is ecological succession? Ecological succession is a process that describes how the structure of biological community over time, like and interacting groups of various species in a desert, forest, grassland, marine environment. And it's three types of primary succession, secondary succession, and serial stage. So next slide. Primary succession. Primary succession is the succession that starts in lifeless areas such as regions devoid of soils or the areas where the soil is unable to the sustain life. When the planet was the first formed, there was no soil on Earth. The Earth was only made up of rocks. These rocks were broken down by microorganisms and eroded from soil. The soil then becomes the foundations of plant life. These plants help the survival of different animals and progress from primary succession to the climax community. Next slide. Secondary succession. Secondary successions occurs when the primary ecosystem gets destroyed, like a climax community gets destroyed by fire. It gets recognitions after the destruction. This is known as secondary ecological succession. Small plants embrace first, followed by larger plants. The tall trees block the sunlight and changes the structure of the organism below the canopy. Finally, the climax community arrives. And serial stage, a serial community is replaced by the subsequent community. It consists of simple food chains. It exhibits a very low degree of diversity. The individuals are less in number and the nutrients are also less. So next slide. Some example in Acadia National Park. These national parks suffered a huge wildfire. Restoration of the forest was left to nature. In the initial year, only small plants grew on the burned soil. After several years, the forest showed diversity in tree species. However, the trees before the fire were mostly evergreen, while the trees that grew after the fire were decided years in nature. Ecological succession of coral reefs. Small coral polyps colonize the rocks. These polyps grow and divide to form coral colonies. The shape of the coral reefs attracts small fish and crustaceans that are food for the larger fish. Thus, a fully functional coral reef exists. Causes. The main causes of ecological succession include the biotic and climatic factors that can destroy the populations of an area. Wind, fire, soil erosion, and natural disasters include the climatic factors. And importance, ecological successions is important more for the growth and development of an ecosystem. It is initiates colonizations of a new area and recolonizations of the area that had been destroyed due to certain biotic and climatic factors. Thus, the organisms can adapt to the change and learn to survive in a changing environment. Thank you, sir.
থ্যাংক ইউ শুভদীপ। ভালো ভালো হবে, করতে হবে, কোনো কিছু পড়ে তারপর প্রেজেন্টেশনটা দিতে হবে, না হলে দেখে দেখে রিডিং করলে প্রেজেন্টেশনটা অতটা ভালো হয় না। আরো ভালো করার চেষ্টা করিস।
রিডিং করাটা কোনো কাজের কথা নয় কিন্তু, জানো তো, রিডিং করবে না, তোমাকে নিজে থেকে বলতে হবে বাবা। ওকে
ওকে স্যার, ওকে ম্যাম।
শুভদীপ, তোমার এই কনটেক্সটেই আমি একটা কোশ্চেন করতে চাই। তুমি ক্লাইম্যাক্স ভেজিটেশনের নাম শুনেছো? ক্লাইম্যাক্স ভেজিটেশন
ক্লাইম্যাক্স কমিউনিটি,
এই সাকসেশনই তো আছে।
ক্লাইম্যাক্স কমিউনিটি আছে।
কমিউনিটি, হ্যাঁ হ্যাঁ, বলো বলো কমিউনিটি। ওকে ওকে, ক্লাইম্যাক্স কমিউনিটি বলো। হোয়াট ইজ ক্লাইম্যাক্স কমিউনিটি?
স্যার, যখন কোনো একটা বিশেষ প্রজাতির সংখ্যা অন্য একটা প্রজাতির সঙ্গে মিশে একটা ওই কমিউনিটির সংখ্যা একটা শিখরে পৌঁছে যায়, ওই কমিউনিটি শেষ পর্যন্ত টিকতে থাকে, তখন ক্লাইম্যাক্স কমিউনিটি।
ওকে শুভদীপ, থ্যাংক ইউ, ভালো প্রেজেন্টেশন, ভালোভাবে ঠিক আছে। নেক্সট সেম 2 থেকে সমৃতা সিনহা ফ্রম সেম 2। সমৃতা, yes.
Yes, sir. Am I audible?
Yes, Somrita. Yes, you are audible.
Am I audible, sir?
Is your slides are visible to you? Visible?
Yes, sir. Yes, sir.
Then do continue.
Good evening to respected principal, ma'am, all departmental teachers, friends, and seniors. I am Somrita Sinha. I am going to present and speak on the topic mycorrhiza and its application. Next slide. Mycorrhiza are mutualistic symbiotic associations between higher plant roots and fungi. Italian mycologist Botany in 1842 was the first to recognize the possible beneficial role of fungal mycelia, which mantle the root of higher plants, and this associate mycorrhiza. Greek word myc means fungi and rhiza means roots. Fungal roots was discovered by the German botanist A.B. Frank in 1855. Next slide, sir. On the basis of morphological and anatomical features, mycorrhiza are divided into 3 types: endomycorrhiza, ectomycorrhiza, and ectendomycorrhiza. Endomycorrhiza further has 5 types: Bam fungi, orchidoid mycorrhiza, ericoid mycorrhiza, orbitoid mycorrhiza, and monotropous mycorrhiza. Next slide, sir. Endomycorrhiza are present on root surface as individual threads that may penetrate directly into root hairs, other epidermal cells, and into cortical cells. Vamp fungi, vesicular arbuscular mycorrhiza, is a type of endomycorrhizal association where both vesicles and arbuscles are developed together. The arbuscles are repeated dichotomously branched hausteria which grow intercellularly. This type of association was present very early in the evolution of land plants. Next slide, sir. Orchidoid mycorrhiza. This association, probably pseudomycorrhiza, play an important role in establishment of orchid seedlings. Ericoid mycorrhiza. They are found in different members of Ericaceae. The fungi are slow-growing, septate, and mostly sterile. Next slide, sir. Orbitoid mycorrhiza. They are variants of ectomycorrhiza found in certain plants by ivy coils in epidermal cells. Monotropoid mycorrhiza. They are belongs to Basidiomycina. It has short fleshy roots that are invested with a hyphal sheath and often forming vertex net in the cortical zone. Next slide, sir. Ectomycorrhiza. They are commonly called seeding mycorrhiza. The fungus enters the cortex forming vertex net but never goes inside the endodermis or stele. Ectendomycorrhiza. This fungi begins with formation of a vertex net which grows behind the apical stem of the growing root. Next slide, sir. Application of mycorrhiza in agriculture. In nutrient deficient soil, the mycelial association helps in the absorption of sodium, potassium, calcium, magnesium, and other elements, produce the germination of seedlings, stimulate the growth of beneficial microorganisms, increase plant resistance to disease and droughts. In forestry, mycorrhiza plays an important role to establish forests in unfavorable locations. Mycorrhiza mix nitrogenous compounds such as nitrate, ammonia, etc. Thus, it helps in plant growth in acid soil. Increased diversity of plants. Trees with facultative endomycorrhiza act as invader of wastelands as pioneer in plant succession. So, I do hereby conclude that mycorrhiza association is very essential for the plants because it has several benefits like absorption of nutrients, enhance plant efficiency in absorbing water and nutrients from soil. Swamp fungi are very useful in the agriculture because it acts as biofertilizers as it helps in the absorption of phosphorus and other nutrients uptake. This is my speech end. I would like to express my deepest gratitude to my mentor, Dr. Debashree Gupta ma'am, and the convener of this seminar, Dr. Argha Sarkar sir, and I'm so much thankful to my HOD, Dr. Orpita Swarnakar ma'am, and Honorable Principal ma'am, Dr. Himadri Chakraborty Bhattacharya. Thanks to my all departmental teachers, friends, and family for their continuous help and cooperation for the completion of this presentation. Thank you.
Very good.
Very good presentation.
সমৃতার স্লাইড গুলো ভারী সুন্দর, বেশ সুন্দর করে সাজিয়েছে।
Well done, Shweta. All of us, your presentation is too good, and presenting the topic is also very good. Now it's time for interaction session. If any question from anyone, please, or from the
Can you name one endomycorrhiza or VAM mycorrhiza? VAM. Can you name one VAM species?
ম্যাম আরেকবার বলুন, আমি শুনতে পাচ্ছি না।
Can you name one VAM fungus? একটা ফাঙ্গাস যে ভ্যান অ্যাসোসিয়েশন করে।
ফাঙ্গাস নেই।
ফাঙ্গাস অ্যাসোসিয়েশন করে এরকম কোনো ব্যাম এর নাম বলতে পারবি? ব্যামের একটা এক্সাম্পল দে।
করতে হতো এগুলো, গ্লোমাস, গাইগাস্পোরা অনেক আছে।
ওকে, হ্যাঁ ইয়েস ম্যাম, গ্লোমাস।
ঠিক আছে, আমাদের বইতেই আছে, হাইতেই আছে, আমি বাইরে থেকে কিছু বলছি না।
ইয়েস ম্যাম। ওকে ম্যাম,
ঠিক আছে।
ঠিক আছে, সঙ্গীতা একটু পড়ে নিবি, হ্যাঁ, যেগুলো সম্বন্ধে প্রেজেন্টেশন পড়ে নিও।
ইয়েস ইয়েস।
এই অর্ঘ্য, আমি অলিদি বলছি, শুনতে পাচ্ছো? আমার আমার ইয়েটা খুব লিংকটা খুব ডিস্টার্ব করছে, দেবশ্রীকে রেকর্ড করতে পারলো, এই সমৃতা অবধি করলাম।
আমি করছি দিদি, চিন্তা করবেন না,
ঠিক আছে। মাঝে মাঝে লিংক চলে যাচ্ছে, খুব অসুবিধা হচ্ছে।
হ্যাঁ আমি আছি, আমি
এতক্ষণ করেছি, এখন আর খুব ডিস্টার্ব করছে বলে। ওকে ওকে।
Okay, so our next presentation, Shomjit model. Shomjit model from Centu. Shomjit from Centu. Shomjit. সৌম্যজিৎ শুনতে পাচ্ছ? সৌম্যজিৎ তো এখানে নেই। সৌম্যজিৎকে আমি এখানে দেখতে পাচ্ছি না, এখনো পিপল লিস্টে সৌম্যজিৎ কানেক্টেড নয়। ওকে, আমি সৌম্যজিৎকে স্কিপ করে নেক্সট প্রেজেন্টেশনে চলে যাব। নাও ইটস টাইম ফর শ্রীপর্ণা। শ্রীপর্ণা সেনগুপ্ত ফ্রম সেন্টু। শ্রীপর্ণা আছো? Are you there?
Yes, sir.
Okay, then turn on your video. Yeah, Shreeparna, wait. Is it visible to you?
Good evening, everyone. Yes, okay, it's visible.
Good evening, everyone present here. I am Shivana Sengupta of Semester 2 Botany Honors, and today for the student seminar, my topic is cell types and evolution in teratophytes. Next slide, please. The teratophytes are the first vascular plants to have the stele. The central vascular cylinder of the primary axis of teratophyte is usually referred as stele, and it consists of xylem, phloem, pericycle, medullary rays, and sometimes peat. The stellar theory was proposed by Van Peegam and Julliard in 1886, and according to the theory, the roots and the stem has some basic structures consisting of the two fundamental units, the cortex and the central cylinder. Next slide, please. In 1982, Smith recognized the two principal types of stele in teratophytes. They are the protostele and the siphonostele. The protostele is the most simplest kind of stele, as it is the simplest and the primitive kind of a stele. It has it is divided into four subunits. They are the haplostele, the actinostele, the plectostele, and the mixed stele. The fossil xylophytals, such as Rhinia, is an example of stele, as well as many primitive living vascular plants like Xylotum is also an example of the protostele. Next slide, please. Haplostele. Haplostele is the first type of protostele, and it is the most simplest and the primitive kind of protostele. Here, the central solid smooth core of the xylem remains surrounded by a ring of phloem. The haplostele is found in fossils like the Rhinia, as well as in many living pteridophytes such as Xylotum. The actinostele. The actinostele is a modified version of the haplostele, as because here the central core of xylem has the radiating rib. The living form of the actinostele is xylotum, and the fossil form is the astroxylem. Next slide, please. The plectostele. The plectostele is the most advanced protostele. Here, the central core of the xylem is divided into a number of plates arranged parallel to each other. It is found in the aerial shoots and the cone axis of Lycopodium clavitum. Next comes the mixed protostele. The mixed protostele, here the xylem elements are mixed up with the parenchymatous cells of the pit. It is found in the stems of Lycopodium cernum. Next slide, please. Now, after the protostele comes the siphonostele. The stele with a pith at a center is usually known as the siphonostele. Regarding the origin of siphonostele, there are two theories. That is the intrastellar origin of the pith and the extrastellar origin of the pith. The interstellar origin of the pith is widely accepted, whereas the extrastellar origin of the pith is not widely accepted because there are many pteridophytes where there is stele without the leaf gaps and having pith in the center. Next slide, please. Now, there are two main theories regarding the evolution of the siphonostele from protostele. They are the intrastellar origin and the extrastellar origin. The intrastellar origin, according to this theory, the siphonostele is evolved by a conversion of the central mass of the xylem into, and according to the extrastellar origin, uh, the pith is originated as a result of inversion of the parenchymatous cell of the cortex into the stele. Next slide, please. And based on the position and the distribution of the phloem, there are two types of siphonosteles. Mainly, they are the ectofluic siphonostele and the amphiphoic siphonostele. The ectofluic siphonostele is usually found in equisetum and others, and the amphiphoic siphonostele is usually found in Dryopteris. The amphiphoic siphonostele is having two endodermal layers, that is the outer endodermis and the inner endodermis. Next slide, please. Then comes the solonostele. A siphonostele which is perforated at the place of the origin of a leaf trace is usually known as solonostele. The solonostele is divided into 6 types. They are the ectophic solonostele, the phloem that is present only at the outer side, example in Osmanda. The amphiphoic solonostele, the phloem which is present on the both sides of the xylem, example aginatum peratum. Next slide, please.
Next comes the dictyostele. The solenoid that is broken into a network of separate vascular strands is known as dictyostele. Each such separate vascular strands is known as the meristle. It is found in serous. Next comes the polycyclic stele. The polycyclic stele, this is it's a bit different. Why it is different? Because here the stele is present or more concentric cylinders. If in a polycyclic stele the outer cylinder is solenoid, then it is called the polycyclic solenoid. And if the outer cylinder is dictyostele, then it is called the polycyclic dictyostele. It is found in Matonia pectinata. Next comes the U stele. This is usually found in the this kind of stele is usually found in the higher plants. It says that if the stele is split into distinct collateral vascular bundles, then it is called the U stele. It is usually found in the dicot stem primary structures, and the last is the attractostele. It is also found in the higher plants. It is similar to the U stele, but the only difference here is that the individual vascular bundles are scatterly distributed into the ground tissue. And the example of this is monocot stele. The reason why I mentioned at the very beginning is that the stereotypes and evolution of stereotypes is because that in gymnosperm the U stele is mainly found, but in pteridophytes here yes sir.
Already
Yes sir.
time. Okay, so conclude very fast.
I'm concluding. Yeah. So in pteridophyte, each and this kind of series, each and every kind of series is being seen. So that's the end of my presentation. Thank you.
Okay, thank you. It's not very good presentation, and the slides were very good, and the topic was good. Your description was
very good presentation.
Slide also very good.
Thank you, sir. Thank you, sir.
Sriporna, I have a question. Which one is the highest evaluated form of stele, and where is it found?
So the U stele.
U stele. Where is it found?
In the gymnosperm.
Highest form in gymnosperm. My question is, which one is the highest evaluated form of stele, and where is it found?
Highest form of evolution has been occurred in which type of stele, and in which type of plant it is. Obviously, advanced group মানে তো সেটা
advanced group
মানে সেটা তো আরো অ্যাডভান্স প্ল্যান্ট হতে হবে।
আর অ্যাডভান্স প্ল্যান্ট হতে হবে। Which one?
Sriporna, for knowledge, angiosperm কোথায়?
Yes, angiosperm.
Yes.
Yes.
Angiosperm, and within angiosperm it is monocot. Monocot.
Yes, sir. Monocot. Yes, sir. I'm extremely sorry.
Okay. So which? Yes. What is the name of that stele?
The last one, which is Actitus.
Yes, attractostele. Yes. Okay, attractostele. Yes. Okay.
Okay, sir. Thank you, sir.
Okay,
so can I
good introduction and
turn off the video and the audio? Okay, sir. Thank you, sir.
Is Shomojit is there? Shomojit Mondal, have you joined? Still not. Then I have to cancel his candidature as a presenter. Then Shuprakash Vishwas from St. Six. Shuprakash. Shuprakash. Audible. Turn on your video.
Sir, I will be presenting.
Yes, first. Yeah, okay. You are also visible and you will present. Sure. So I stop my presenting. Yeah, start your presentation.
Is it visible,
Yes,
sir?
clearly.
Yes, yes.
Good evening, everyone. My name is Shuprakash Biswas, and I'll be talking about protein. So, what is protein? Proteins are large biomolecules and micromolecules which are comprised of one or more long chains of amino acids. As you can see that amino acids consist of amino group, a carboxylic group, a central carbon, and a side chain which is unique to each amino acids. It consists of carbon, nitrogen, oxygen, and hydrogen. Moving on to the next slide, we can find different types of proteins and functions. In animals, the digestive enzymes such as amylase, lipase helps in digestion of food. Transport proteins such as hemoglobin, which carry substances in the blood throughout the body. Structural protein such as actin, tubulin construct different structures like the cytoskeleton. Hormones such as insulin and thyroxine coordinate the activity of different body systems. Immunoglobulin, which is the antibiotics in our body, which helps in foreign pathogens.
Antibodies.
Antibodies, which helps our body from foreign pathogens. Moving on to the next slide. There are 20 different kinds of amino acid which can be found in our body or we can get from the food. So there are actually 5 non-essential amino acids which we can get from foods or we can make ourselves, like alanine, asparagine, asparagic acid, glutamic acid, and serine. There are five conditional five conditional essential amino acid which are which can be made by healthy bodies, and we can't make them in starvation or in inborn error of metabolism, like arginine, cysteine, glutamine, glycine, proline, tyrosine. And there are 9 essential amino acids which we can only get from food, like histidine, isoleucine, leucine, lysine, and others. So we can see that there are different structure of proteins. There are mainly 4 kinds of protein structures. The primary structure is a linear arrangement of amino acid which is linked by peptide chains. The secondary structure is consisted of 2 types of structure, that is alpha helix and beta plated sheet, and they are joined together by peptide bonds and hydrogen bonds. Tertiary structure is the polypeptide chain, which is in a spatial arrangement and is consist of only one chain, whereas quaternary structure consists of more than one chain. So we can find various roles of protein in plants, such as we all know that enzymes are proteins, and therefore enzymes are present in different metabolic pathways in our plants, such as glycolysis, TCA cycle, C3C4. In our cell membrane, we can find globular proteins such as integral protein and peripheral protein. Structural protein helps to form our backbone of the plasma membrane and impart mechanical strength to the cell. Carrier protein such as uniport, symport, and antiport helps in transfer of substances across the cell membrane. So how protein is utilized in our body? We with the protein we take have essential amino acid which helps in making proteins, hormones, and other important molecules in our body. The proteinaceous food that we eat goes into the stomach, and we digest it with the help of HCl, which helps in denaturing of the protein. And the denatured protein is acted upon by pepsin, which is produced by gastric cell, and it breaks into various different kinds of chains and different kind of oligopeptide chains. This oligopeptide chains is then transferred to duodenum, Here it is acted by different kind of digestive enzymes. It then further gets degraded into tripeptides, dipeptides, and individual amino acids. It then get transferred to intestinal cell where there are various kinds of enzymes, and the some of the amino acids get synthesized in the intestinal enzymes and new cells, and most of the amino acid enters the bloodstream, which are transported to the other parts. Moving on to the next slide, there are daily requirements of protein in our body, and it varies for varying age. Like from age 1 to 3, they require 13 grams of protein. From age 4 to 8, they require 19 grams. From 9 to 13, they require 34 grams of protein. The protein varies for female and male. For female, it is 46 grams, and for male, that is 52 grams. For an adult male, which they require 56 grams of protein, and we can see that proteins vary from different age group and different physical activities. Like the sportsmen require more protein so that they could carry out um, e- they carry out energy, and the old persons can also benefit from protein, as they will require more amount of it to get the life going. So let's recap. There are 5 non-essential amino acids. There are 6 non-conditional essential amino acids. There are 9 essential amino acids which we can get from our diet. So protein needs vary depending on various life cycle stage and health status. Some health conditions require um, uh, some health conditions require more requirements of protein, or if we don't consume more protein, we can be even deficient. Everyone can meet protein needs through eating a variety of foods, whether it is omnivorous, vegetarian, or vegan. So these are the resources which I have gathered the information. Thank you for such a wonderful seminar. Thank you to my teachers, our HOD, Dr. Arpita Swarnakar, my mentor, Dr. Debashri Gupta, and all of my teachers, my students, and everyone. Thank you.
Thank you, Shuprakash. Though it is slightly out of time, already it's 5 minutes exceeded. It's 2:06, but presentation is good and the slides are excellent.
Slides are really
good. This is the information for all of the students. All the slides should be informative, not the descriptive. স্লাইডে সবসময় ইনফরমেশন দাও অল্প করে এবং সেটার সম্বন্ধে নিজে এবার এক্সপ্লেন করো, এটাই হচ্ছে আপনার প্রেজেন্টেশন। In that context, these slides that described by Shuprakash is very good. But one thing, Shuprakash, I would say, and that is the topic was not at all protein for yours. It was a biomolecules or molecules of life, and you have sorted out one of this. But my suggestion is, from later times, do stick to the point only that is supplied or provided to you. When you are supplied the molecules of life or biomolecules, then you have to cover all those things. You have to cover protein, you have to cover carbohydrate, lipids, and any other biomolecules that exist in life. So, yes.
No, actually, he asked me. Then I told him that will be too big for him to cover in 5 minutes. So better choose one of the topics and do it. That was my suggestion to Shuprakash.
Yes, that your solution, that's very fine. That's very nice. But that my assumption is, as biomolecules are molecules of life, when we'll go through, then only the introductory points.
He could have said in the beginning that the biomolecules, how many types of biomolecules are there, and he is selecting one of them.
Right.
That he could have said in the beginning.
Otherwise, this is a very good. Okay. So, any question for Shuprakash from the interaction session? Biplab da Aditi, even Devashri, if you have any question for Shuprakash.
আচ্ছা সুপ্রকাশকে আমি একটা বলি, সুপ্রকাশ, হোয়াট ইজ দ্য
Yes,
what
sir.
is the difference between structural protein and functional protein?
So structural proteins are those kind of protein which are there in cell membrane, and that helps in the structural, like the cytoskeleton of the cell membrane. And the functional protein are those kind of protein which can be seen in various kinds of cells, and they can also be in the form of enzymes.
Yes, that is the for functional purpose. Okay. Yes. Okay, good.
Okay, thank you, Shubprakash. Now you can stop your presentation. So there is a last one participant, Vishal, but Vishal is not there. He is trying for many times, and he has texted me that he is trying for joining, but network issue is so much there from his end, according to him. So Shubprakash is the last contestant as a participation. Is the last one that Shubprakash has made it. So all the participation has been already done. Shomodis is not there. So we had 25 participants for presentation, but 23 have participated and presented their topics. Only 2 were absent. One is Shomojit Mondol and another is Vishal Poddar. Shomojit Mondol is from SEM 2, Vishal Poddar is SEM 4. I would request our evaluators only to note those 2 names, Shomojit Mondol for SEM 2 and Vishal Poddar for SEM 4 as marked them absent.
Yes, we have done it.
So now it's time for the sum up the whole things. It's a long time. It's about 8 p.m. So about 4 hours we have already passed through for this lovely presentation, and very thanks to all of you guys. These are very nice experience that we have experienced today, and whatever the performances that you have already did by the means of your deliberation, that's really very good, and it is our beyond of expectation and imagination that you have made it up. So again, thumbs up to all of you. Now it's time for the total matchup and total declaration of the views that our evaluators, Dr. Devoshri Gupta and Dr. Vadi Kanchan Madam, has collected, and they will just sum up their views, and they will share the views, and they will give some suggestions for you, so that suggestions can be beneficial for your future goals. Okay, so over to Olidi and Devoshri, please.
Olidi, are you there?
Olidi আছেন? ছিলেন তো?
Olidi তো নেই, মনে হচ্ছে Olidi লিভ করেছেন বোধহয়।
নেটওয়ার্ক ইস্যু হচ্ছে। সো Olidi জয়েন করলেন?
Olidi জয়েন করেছেন।
হ্যাঁ দিদি, শুনতে পাচ্ছেন না? আপনাকে আপনি আনমিউট করে নিন দিদি। Olidi আনমিউট করে নিন।
হ্যাঁ করেছি, আনমিউট করেছি। খুবই অসুবিধা হচ্ছে, লিংক চলে যাচ্ছে বারবার। হ্যাঁ এবার করেছি,
এবার আমাদের টাইম
আনমিউট করেছি।
আপনারা আপনাদের সাজেশন বা ভিউজটা কি বলেন, আপনি আগে বলুন, তারপরে দেবশ্রী বলবেন।
আমার যে লিংক চলে যাচ্ছে,
আপনি ভিডিওটা অফ করে বলুন দিদি, ভিডিওটা অফ করুন।
ভিডিওটা অফ করে দিই।
হ্যাঁ সেটা ভালো হবে।
হ্যাঁ এক সেকেন্ড। আমার মনে হয় ইট ওয়াজ এ ভেরি এক্সেলেন্ট সেমিনার। মানে আমি ভাষায় প্রকাশ করতে পারছি না যে এত সুন্দর অনলাইনে দ্য স্টুডেন্টস হ্যাভ পারফর্মড ভেরি ওয়েল। কেউ বেশি কেউ কম, এ তো হবেই। হ্যাঁ, আর যার যা ঘাটতি ছিল আমরা দেন বলে দিয়েছি, কার স্লাইড ঠিক হয়নি বা কেউ কোন পোর্শনটা সে ভালো করে বুঝতে পারেনি, আন্ডারস্ট্যান্ডিং কম ছিল বা প্রেজেন্টেশন কম ছিল, সেগুলো আমরা দেন বলেই দিয়েছি। তা আমার যেটা বলার, স্টুডেন্টস ইউ নট অনলি ইন দি ডিপার্টমেন্ট, যেখানেই সুযোগ পাও ইউ কিপ participating in such type of programs which will help you in your future life to conduct to face different examinations, different interviews, or for higher studies. And best wishes to all of you, and also the organizers, and also to our principal madam and TC secretary. Thank you all, head of department, etc. এই আর আমি বেশি কিছু বলছি না, লিংক চলে যাচ্ছে বারবার।
ওকে দিদি,
দেবস, হ্যাঁ
হ্যাঁ বলছি যে it was really a wonderful occasion to meet you all, and it was really wonderful to hear from you. All you all people did wonderfully well. I just can't imagine that we at this age could do all these things. So
that's correct.
Yes, and that I can't imagine. So this is really formidable job you have done. So some of you have fared less good, some of you fared better, some of you fared best, but that is always the way of competition. Okay, so you don't have to worry if you get a first position or you get second position. You don't worry about that. You keep on participating. And one thing I want to tell Argha, that if possible, we will try to convene this kind of seminars when we can include all the students of the class, because some are feeling left out. So they couldn't qualify because of their write-up, but they were good students. So we can do that one thing, that one day we can do with the semester, one semester, and then on the other day with other semester. That will be little painstaking, but that will help the students, I think.
I think also this is exactly the right proposal that you have created, that we should inculcate all those students though as already not participated. Yes, they can feel morose that we have not participated, but yes, if we can plan.
And everybody needs practice. Everybody needs the practice.
If we can plan such a way that in every week for one semester is purposely done, then that would be done.
That we can accommodate, I think. That we can do. We can do.
In that case, time constant will not be the factor.
Yes. Thank you, everybody. Thank you, Argha. Thank you, Orpita. Thank you, Olidi, Rakadi, Pushpitadi, and our principal madam, and Biplav. Everybody, thank you. And TS sir, of course, I think he is there. I don't know whether TS sir is there or not. Okay, I thank him also, and most of all, I thank all my students. Well done, all of you.
Yeah, thank you, Didi. And this is a very good time that we have already spent, and Diji Madam has already said a lot about you that you have already presented, and exactly we will witness some more enthusiastic, such significant events where you will participate, you will the monitors, and you will handle those batons. And exactly if these batons are being run by our semester students, then only we can also propose this type of events in intercollegiate level. Our principal madam has already said me when I just did a proposal to him, then he suggested that do some job much more in case of intercollegiate when other few college students can join the seminar. So we will try to organize a seminar if it is possible. Then it would be a very much proud factor for us also. So thank you, students. But one thing that you have to know, that to get all those certificates, you have to fill the feedback form. I thought that I would share the feedback form link here, but so many of them had left due to the network issue. Again, the time is also very much constrained. The 4 long hours have been spent. So I would post the link to your individual groups, and you will put your inputs very minutely and sincerely in the feedback form, because the feedback form that you will put the information, according to that, on the basis of your information, the certificate will be generated, and this certificate will be dispatched to you by your email ID. And this will take time, as because the score sheet will be finalized by the evaluators, and after that also the ranks that you have already secured, the ranks will be announced later.
Okay, let me ask you one thing, Argha. Ranking, what we are giving ranking? First, second, third? Or
No,
only
no,
first?
second, third. 3 prizes.
Oh.
And 3 certificates will be nominated for first, second, and third respectively. And obviously, the participating certificates will be available for all the students. So just minutely and sincerely fill up the forms. In case of filling the feedback form, one thing you have to keep it in mind: there is a last column that is a title of presentation. Who have presented their topic? Just write the topic there. It is not for the participator who have not presented. It is only for the presenter column. Okay, so that is not a mandatory column, but you have to fill who have presented, because the title or the topic that you have presented, if it is mentioned in your certificate, that would carry the credential. Okay, so thank you so much. I will give the feedback link to your group. Now it's a time of vote of thanks. I will request our very own Dr. Biplab Bagchi to say the vote of thanks and to complete the session. Biplab.
At first, I will give my a big thanks to Dr. Argha Sarkar. He has proposed this seminar and acted as a convener successfully. Next, I will thank our principal, Madam Dr. Himadri Chakraborty Bhattacharya. She was present here and gave valuable time. I will give her a big thanks. I also I convey my heartfelt thanks to Dr. Pushpita Sarkar, our Teachers Council Secretary. She has spent her valuable time for this seminar, and I will also give great thanks to our departmental head, Orpita Di. She has supervised overall the seminar as HOD, and I will give my handful thanks to Devasridi and Olidi, who has acted as judges here, and also mentor all the teachers who acted as mentor for the development of the students, how to prepare their slides, and how to develop their views and ideas about the seminars, about their thought topics. They have given their valuable suggestions. So at last, I will give great thanks, large thanks, and also many claps to our beloved students. They have performed very well, although it was first time for a deliberation in PowerPoint presentation. I know it is very hard work to prepare PowerPoint presentation in Android mobile. It is easy, we know it is easy in laptop and desktop, but it is very hard to in mobile. But they have performed this hard work. Next time, all students have done very well. Next time they will do more and more good. Okay, thank you everybody.
So thank you, Biplab. So thanks to everyone. So seeking permission from HOD Vidati and seeking permission from all of our seniors, Devasridi, Biplab, Olidi over there, and Raka, I think not is here. So I would just complete here, and I will terminate the session. Thank you, students, beloved students. Take care of your own health, relatives, and your bodies. Bye. Thank you.
Argha, I would like to thank all of you and the teachers and the students also, because they have strived so hard for this. They have prepared excellent slides and they have presented, and I'm very glad that how they learned, and they have prepared the PowerPoint slides on a mobile phone, which is still very difficult for me as well, but they have done it. So kudos to them. And I think in the upcoming examinations, and especially internal examinations will start from 19th. So they will prepare very hard and show their good results over there also, like they did today. But to me, I think everybody who has participated or tried to participate at least, they are all winners. And I think they should be given saluted for that. So keep on trying and this should go on. So thanks to all of you, all my colleagues and students once again. Thank you.
So thank you, students. I'm just completing the recording. Stop the recording and stop the program. Thank you so much.
Thank you.
Thank you, teachers.
Thank you, sir.